An audiobook rendering of Mechanistic Insights into Stomatal Regulation Under Water Stress by Sahil Desai. A dissertation. Presented to the faculty of the Graduate School of Cornell University in partial fulfillment of the requirements for the degree of Doctor of Philosophy by Sahil Desai. August 2026. Copyright 2026. Sahil Desai. All rights reserved. Abstract. Stomata, the microscopic pores that govern more than 95% of terrestrial water and carbon exchange, set the central trade-off between photosynthetic carbon gain and transpirational water loss, making them a decisive control point for crop water use efficiency under a warming, increasingly water-limited climate. Yet despite decades of study, stomatal physiology has lacked a unified mechanistic account of how leaf hydraulic transport, guard cell ABA signaling, and outside xylem resistance are integrated during water stress. This dissertation addresses this gap by combining direct in-planta measurements of water potential with mechanistic modeling spanning the guard cell to the soil. Using AquaDust, a fret-based nanoreporter that resolves water potential upstream and downstream of the outside xylem zone, together with genetic suppression of hydroactive regulation in SLAC1 mutants, we partition stomatal regulation into its hydropassive and hydroactive components. In maize, we confront a hydropassive model with drought stress response and find that it is sufficient to reproduce stomatal response in the SLAC1 mutant, where active regulation is absent, but not in the wild type, where hydraulics alone cannot account for the response and hydroactive regulation is required. Across both genotypes, the ratio of stomatal to outside xylem conductance, GS divided by GOXZ, recently established as a phenotype governing non-stomatal control of transpiration in maize, reaches 0.3 to 0.5 under moderate drought, driving airspace undersaturation that causes standard gas exchange measurements to underestimate stomatal conductance by 20 to 25%. We then develop a coupled hydropassive hydroactive model in Arabidopsis, showing that guard cell autonomous ABA autoregulation provides the core nonlinearity that generates both stomatal oscillations and the hysteretic stress memory observed across drought and recovery. Extending the AquaDust and GS divided by GOXC framework to cotton reveals a sharp C3. C4 contrast. Cotton exhibits measurable airspace undersaturation, yet keeps GS divided by GOXC on the order of 0.01 and remains stomata-dominated, in contrast to the strongly non-stomatal regime of maize. Finally, confronting a one-dimensional transient soil drying model against direct soil water potential measurements down to negative 30 megapascals demonstrates that standard soil hydraulic parameterizations fail in the extreme dry regime relevant to severe drought and may therefore misplace the upstream boundary at which plant water stress first arises in the soil-plant atmosphere continuum. Together, these results connect cell-scale biophysics to whole plant and soil hydraulics, establishing psi resolved measurement as the basis for a mechanistic, quantitative, and predictive account of stomatal regulation and a foundation for engineering crops resilient to water stress. Chapter 1. Introduction. Section 1.1. Stomata, Water, and Global Food Security. Stomata are microscopic pores on the leaf epidermis that regulate the exchange of water vapor, H2O, and carbon dioxide, CO2, between terrestrial vegetation and the atmosphere, accounting for more than 95% of all terrestrial gaseous flux of water and carbon. Agriculture currently accounts for roughly 70% of all freshwater withdrawals worldwide, and climate projections consistently indicate that drought frequency and severity will increase through the 21st century. Improving crop water use efficiency, WUE, defined as the ratio of carbon assimilation, A, in units of mu mole per meter squared per second, to transpiration water loss, E, in units of millimole per meter squared per second, is therefore a central challenge for food security under a warming and water-limited climate. Stomatal aperture governs the critical trade-off between CO2 uptake for photosynthesis and water loss via transpiration. Both fluxes are regulated by the stomatal resistance, that is, 1 divided by GS, making stomatal conductance, GS, measured in millimoles per square meter per second, a crucial determinant of plant productivity, WUE, and drought resilience. In the soil-plant atmosphere continuum, or SPAC, water potential, PSI, in megapascals decreases from the soil, soil, through the xylem, xyle, and the leaf apoplast at the evaporating surface of the substomatal cavity, SSC apo, to the atmosphere, air, driving water flow while the stomatal resistance sets the A-E trade-off at the pore, a gamble in Graham Farquhar's words. Despite decades of research, stomatal physiology still lacks a unified mechanistic account of how water status, hydraulic transport, and biochemical signaling are integrated during water stress. In particular, existing frameworks have not fully resolved how hydraulic architecture at the leaf scale, guard cell, ABA biosynthesis and signaling, and outside xylem resistance interact to determine stomatal behavior across species and environments. This gap limits the ability to explain variation in stomatal responses and constrains efforts to predict or improve crop water use efficiency under increasing water limitation. A clearer mechanistic understanding of this regulation is therefore needed to connect plant-water relations to crop performance and global food security. Section 1.2, the Soil-Plant Atmosphere Continuum, SPAC. Water movement through the SPAC is driven by gradients in water potential, PSI, measured in megapascals, with psi decreasing from the soil through the xylem and outside xylem zone, or OXZ, to the substomatal cavity apoplast and then to the atmosphere. Water potential in the SPAC spans an immense range, from near zero values of roughly negative 0.001 megapascals in moist soils to extremely negative values approaching negative 100 megapascals in dry air. Each of these compartments represents a hydraulic node connected by resistances and capacitances that together shape leaf water status. Cowan formalized this perspective by treating the SPAC as an electrical RC network, in which water potential is analogous to voltage and water flux is analogous to current. The plant leaf operates between two boundary conditions: water availability, quantified by xylem water potential CIXYL, and atmospheric water demand, quantified by the vapor pressure deficit, VPD, measured in kilopascals. Jarvis first systematically characterized how stomatal conductance GS responds as a multiplicative function of light, VPD, temperature, CO2, and leaf water potential. This empirical framework is practically useful at canopy and land surface scales, but treats stomata as a black box, without resolving the mechanistic links between environmental drivers, tissue hydraulics, and guard cell biophysics that motivate this thesis. At the stomatal pore, increasing GS raises both carbon assimilation A and transpirational water loss E, and the intrinsic water use efficiency A divided by GS captures this trade-off in a way that is convenient for modeling. However, finite outside xylem, or OXZ, resistance means that stomata are not always the sole limiting pathway for water vapor, a fact that standard gas exchange analyses can obscure because they assume saturated intercellular air spaces. The soil represents the least negative node in the hydraulic continuum. As soil dries, soil water potential seaside soil becomes more negative, and the unsaturated hydraulic conductivity declines by several orders of magnitude as pores empty and the connected liquid network breaks down. Neutron radiography measurements now show that hydraulic limitation often arises first at the root-soil interface, rather than in the xylem or leaf, underscoring soil-water dynamics in the rhizosphere as a critical component of plant drought response. Section 1.3, Leaf Anatomy, Stomatal Complex and Emergent Behaviors, Sum of Stomatal Conductance. A leaf cross-section, illustrated in Figure 1.2a, reveals the pathway that the transpiration stream must traverse, from vascular tissue, that is xylem, through bundle sheath and mesophyll cells, to subsidiary and guard cells in the leaf epidermis, before water evaporates from cell interfaces into the substomatal cavity and exits the leaf through the stomatal pore. The leaf is geometrically divided into an adaxial surface above the xylem plane and an abaxial surface below it, and in C4 species such as maize, the bundle sheath is hydraulically coupled to the mesophyll. In the epidermis, illustrated in Figure 1.2 B through C, stomata are formed by pairs of guard cells flanked by subsidiary cells. The mechanical interaction between the subsidiary cells and guard cells in the stomatal complex is critical for stomatal behavior. Subsidiary cells exert a back pressure on the guard cells so that a uniform decline in leaf water potential can open the pore because subsidiary cell turgor relaxes faster than guard cell turgor. This mechanical advantage of the subsidiary cells relative to guard cells underlies hydropassive responses and provides a structural basis for counterintuitive stomatal dynamics. Between the xylem and the guard cells lies the outside xylem zone, comprising mesophyll symplasm, apoplast, cell walls, and the intercellular vapor space. Standard gas exchange analysis following gastra assumes that the leaf airspace is water vapor saturated, which is equivalent to assuming infinite outside xylem zone conductance and assigning all resistance to the stomatal pore. This assumption was challenged first by Jarvis and Sladier and is definitively disproven by aquadust measurements, which show large drops in water potential between xylem and substomatal cavity apoplast. The ratio of GES to G outside xylem zone therefore quantifies the relative importance of stomatal versus non-stomatal resistance and serves as a mechanistically grounded phenotype for water use efficiency. Guard cells provide an active biochemical response to water stress through the stress hormone abscisic acid, or ABA, signaling, which regulates their osmotic content and turgor. Under declining water status, ABA accumulates in guard cells and triggers ion efflux, reducing osmotic potential and closing the pore. Stomatal behavior under water stress thus emerges from the interaction between hydropassive, or HP, mechanical advantage and hydroactive, or HA, ABA-driven control of guard cell osmotic potential. Together, these HP and HA processes generate the characteristic transient and steady-state patterns of stomatal conductance that define the phenomenological targets of this thesis. Upon a step increase in VPD, GS briefly rises before declining to a new, lower steady state, a transient termed the wrong-way response. The wrong-way response was first modeled mechanistically by Cowan and Delwich and Cook as arising from the phase lag between subsidiary and guard cell turgor responses in a purely HP framework. To overcome this initial wrong-way response and achieve sustained stomatal closure, guard cells must actively adjust their osmotic potential, producing a right-way response that reflects the hydroactive ABA-driven component of regulation. Under moderate step increases in vapor pressure deficit, during closure, G sub S exhibits damped oscillations before reaching steady state. Under high vapor pressure deficit, oscillations can persist indefinitely, as shown in Figure 1.3a. This transition from damped to sustained oscillations is observed experimentally by Farquhar and Cowan, but not yet mechanistically grounded. Figure 1.3b illustrates stomatal hysteresis. Under cycles of dry down and recovery, GS follows different paths for the same psi XYL, so that GS is not a single-valued function of Ci XYL, but depends on the history of water status trajectory. Experiments have characterized this hysteresis and speculated about guard cell signaling components associated with it, but the mathematical and mechanistic origins of the hysteresis itself remain unresolved. Beyond these dynamic behaviors, steady-state GS measurements under drought routinely underestimate true stomatal conductance when internal airspace undersaturation occurs. Jane et al. Showed in maize that GS divided by GOXE can approach 0.3 to 0.5, leading to 20 to 25% errors in standard Li core gas exchange calculations, underscoring the need to characterize this non-stomatal OXE phenotype alongside stomatal regulation itself. Section 1.4. Review of stomatal conductance models. Stomatal models fall into three broad categories that reflect different philosophies about what constitutes a satisfactory explanation of plant behavior: empirical, optimality-based, and mechanistic, or process-based. These categories are not mutually exclusive and address different spatial and temporal scales and purposes, but their cumulative limitations motivate the mechanistic synthesis pursued in this thesis. Section 1.4.1 Empirical Models. Jarvis expressed GS as a multiplicative product of dimensionless response functions for light, VPD, temperature, CO2, and leaf water potential, with each function fitted independently from data and no mechanistic coupling between environmental drivers. The model is widely implemented in canopy scale and land surface models because of its simplicity, but it cannot predict any of the transient behaviors in Figure 1.3 because it has no dynamics, and its parameters are species, site, and season-specific. Ball and colleagues introduced the insight that GS is empirically proportional to the product of net assimilation, A, relative humidity at the leaf surface, and the inverse of CO2 concentration at the leaf surface. This coupling to A was a major advance because it explained the observed correlation between GS and photosynthesis. Ballberry remains the most widely implemented stomatal module in global vegetation and climate models, yet it contains no mechanistic water stress term and is entirely steady state. Medlin and colleagues built upon the Ballberry framework, replacing the empirical relative humidity term with a VPD-based function, providing a more physically interpretable but still steady-state formulation. Looning replaced the relative humidity term in the Ballberry model with a hyperbolic vapor pressure deficit function and introduced the CO2 compensation point, making the model more physically interpretable, but the framework remains fundamentally empirical. Empirical models, therefore, remain effective black boxes. They cannot explain the mechanistic basis of stomatal responses, cannot be reliably extrapolated beyond the environmental and species domains for which they were calibrated, and fail to capture newer findings such as substantial outside xylem limitation of transpiration revealed by aquadust measurements in maize. Section 1.4.2 Optimality Models. Cohen and Farker posited that stomata behave so as to maximize carbon assimilation over a given period subject to a fixed water budget, yielding the optimality condition, the partial derivative of A with respect to E equals lambda. This theory captures the observed tendency of stomata to maintain a nearly constant balance between internal and ambient CO2 across diverse environments without fitting species-specific parameters, but it treats lambda as an externally specified constant with no direct physical basis, provides no mechanism by which stomata achieve the optimum, and contains no dynamics. Catul and colleagues demonstrated analytically that if the marginal water cost scales as atmospheric CO2 concentration, the optimal GS displays the CO2 sensitivity observed empirically, providing a mechanistic rationale for Ballberry-type coupling. Medlin and colleagues reconciled the optimal and empirical approaches to stomatal modeling, showing that the optimal stomatal theory predicts a specific functional form relating GS to A, VPD, and ambient CO2 concentration that is consistent with experimental data. Carminati and Javot used a hydraulic supply function model to show that soil physical constraints can drive convergence in the critical stomatal water potential across diverse species, suggesting that drought responses are strongly shaped by soil-plant hydraulics rather than guard cell biochemistry alone. Despite these insights, optimality-based models remain phenomenological. They encode what stomata should do under given constraints, but lack a mechanistic description of how GS is regulated in real time, and they could therefore benefit from integration with process-based stomatal models that explicitly represent hydraulic and ABA signaling mechanisms. Section 1.4.3 Mechanistic, Process-Based Models. Cowan's landmark paper established the RC network representation of the SPAC, setting up a theoretical basis for the hydropassive, or HP, framework. Delwich and Cook extended this hydraulic framework analytically and identified the parameter regimes in which a purely hydropassive circuit produces oscillations consistent with Figure 1.3a. These foundational HP references locate the oscillatory regimes within the hydraulic circuit, but encode no molecular information about the guard cell processes that set those parameters. Buckley integrated guard and subsidiary cell turgor mechanics, hydroactive feedback via ATP-driven osmoregulation, and the Farquhar-Wong photosynthesis coupling into a single hydromechanical model. It predicted both wrong-way and right-way responses and identified the central role of the guard epidermal turgor pressure difference in determining GS. Buckley and co-authors also demonstrated that HP processes alone cannot explain the magnitude of drought-driven stomatal closure in angiosperms, establishing that HA regulation is required and directly motivating the experimental and modeling work of this thesis. Chen and colleagues and Hills and colleagues developed OnGuard, a comprehensive kinetic model of guard cell plasma membrane and tonoplast transporters. Wang and colleagues extended it to the leaf scale, calling it OnGuard 2, coupling guard cell ion transport to leaf water status and predicting VPD responses without explicit ABA. OnGuard 2 is the principal complementary platform that resolves guard cell electrophysiology at much greater resolution but treats ABA as a prescribed input rather than a dynamically regulated variable. Chapter 3 of this thesis argues that this choice matters because ABA autoregulation, not merely ABA levels, is necessary to explain the oscillations and hysteresis illustrated in Figure 1.3. The most recent OnGuard 3E adds an ecophysiology interface, but retains the phenomenological ABA prescription, leaving the gap that Chapter 3 fills. Peek and Mott demonstrated that GS responds steeply to the vapor pressure difference in the substomatal cavity. Their model proposed that guard cells equilibrate to vapor in the substomatal cavity, near the pore base, providing a vapor phase mechanism for humidity sensing. This mechanism requires near saturation in the substomatal cavity, a condition that aquadust measurements in Chapter 2 show to be violated under moderate drought. Blatt and colleagues have recently proposed that cell wall water buffers guard cells against falling airspace humidity, providing a new perspective on the undersaturation debate that directly engages with the findings of Chapters 2 and 4. Jane and colleagues is the experimental cornerstone of Chapters 2 and 4. By simultaneously measuring Psi-XIL and Psi-SSC-APO in intact transpiring maize leaves, they showed a 25-fold decline in G-oxes, beginning well above the turgor loss point, with Psi-SSC-APO reaching negative 10 megapascals, entirely independently of VPD and stomatal behavior. This VPD independence directly contradicts the evaporative demand-driven undersaturation proposed by Marquez and colleagues and Wang and colleagues, and undermines the vapor phase sensing framework of Peek and Mott. The work of Desai and Stroke that emerged out of this thesis presents the first coupled hydropassive, hydroactive framework that resolves the hydraulic circuit from xylem to guard cell explicitly, couples it to a mechanistic model of guard cell autonomous ABA dynamics, comprising turgor-driven biosynthesis, ABA positive feedback, and ABA catabolism via A8H, CYP707A, and predicts genotype-specific transient responses in Arabidopsis. This model occupies the intersection of the hydraulic and biochemical frameworks, bridging Cowan at one end and the OnGuard family at the other. Section 1.5, Thesis Scope, Organization, and Contributions. The review above identifies 4 unresolved problems that motivate the experimental and modeling chapters of this thesis. First, no study had combined genetically suppressed hydroactive, or HA, regulation with direct in-plant-to-water potential measurement to quantify the purely passive contribution to stomatal regulation. This gap is addressed in Chapter 2. Second, no prior hydropassive hydroactive, or HPHA, model had explicitly resolved ABA biosynthesis kinetics and linked them to both transient behaviors, wrong-way responses, and oscillations, as shown in Figure 1.3a, and steady-state behavior, specifically hysteresis, as shown in Figure 1.3b. This gap is addressed in Chapter 3. Third, the non-stomatal outside xylem, or OXZ, hydraulic phenotype was known only in maize and the characterization of its importance in cotton remained only preliminary. This gap is addressed in Chapter 4. Fourth, standard soil hydraulic parameterizations are extrapolated far beyond their calibration range during severe drought, and direct site-eye soil measurements, down to about -30 megapascals, are needed to constrain them. This gap is addressed in Chapter 5. Chapter 2 presents a direct quantification of stomatal and non-stomatal, that is, OXZ, physiology in wildtype and SLAC1 maize across a drought trajectory, using AquaDust and LI-Core 6800 gas exchange. A purely hydropassive model fitted by Bayesian Markov chain Monte Carlo exclusively to SLAC1 data is then used to infer the hydroactive contribution in wildtype, so that systematic deviations of wildtype data from HP model predictions quantify where and how much active HA input is required. The ratio GS over GOXZ emerges as a mechanistically grounded water use efficiency phenotype that is actionable as both a screening metric and a modeling target. Chapter 3 develops and validates a coupled HP HA model incorporating guard cell autonomous ABA autoregulation, including a stress-driven biosynthesis term, a positive feedback amplification term, and a catabolism term via A8H, CYP707A, alongside the PYR, PP2C, OST1, SLAC1 signaling cascade. The model predicts genotype-specific damped and sustained oscillations and bistable hysteresis in Arabidopsis, establishing ABA autoregulation as the core nonlinearity underlying the dynamic behaviors illustrated in Figure 1.3. Chapter 4 uses AquaDust to characterize the outside xylem zone and stomatal responses in wildtype and GH/SLAC1 cotton across a range of Psi-Axile and VPD combinations. Cotton is found to remain stomata-dominated in sharp contrast to maize, establishing a C3 to C4 species, contrast in outside xylem zone vulnerability, and revisiting, in a direct measurement framework, the debate on the sites of evaporation within the leaf initiated by Jarvis and Sladier. Chapter 5 implements a one-dimensional transient Richards equation model with Van Genuchten water retention characteristics and isothermal Philip de Vries vapor conductivity, confronted against AquaSheet measurements. The chapter shows that standard constitutive parameterizations of soil fail below about -20 megapascals, calling for reparameterization in the extreme dry regime that increasing drought frequency makes agronomically relevant. Chapter 6 synthesizes the 4 experimental and modeling themes and identifies future directions, including the development of species-specific unified HP-HA models, an ABA-OnGuard-HP hybrid with explicit A-GS coupling, extensions to CA2+, reactive oxygen species, ROS, and long-distance signaling, and a coupled soil-root-stomata S PAC model constrained jointly by AquaDust and AquaSheet. Chapter 2. Stomatal and non-stomatal responses in maize wildtype and SLAC1 mutants inform a mechanistic model of hydropassive stomatal regulation. S. Desai, S. Sen, P. Jane, F. E. Rockwell, R. J. Tuhey III, A. J. Studer, A. D. Stroke. Stomatal and non-stomatal responses in maize wildtype and SLAC1 mutants inform a mechanistic model of hydropassive stomatal regulation, in preparation for plant physiology. Author contribution. S. Desai implemented the data analysis, including binning of the SLAC1 data and statistical analysis, developed the hydropassive model, and established the MCMC Bayesian learning pipeline. S. Desai and A. D. Strook analyzed the results and wrote the paper. Stomata are microscopic pores on the leaf epidermis that regulate the exchange of water vapor and carbon dioxide between terrestrial vegetation and the atmosphere, accounting for more than 95% of all terrestrial gaseous flux of water and carbon. Stomatal aperture controls transpiration, that is E, the flux of H2O in millimoles per meter squared per second, and net carbon assimilation, that is A, the flux of CO2 in micromoles per meter squared per second. Both fluxes are regulated by the stomatal resistance, 1 over GS, making stomatal conductance, GS, a crucial determinant of plant productivity, water use efficiency, and drought resilience. In the soil-plant atmosphere continuum, or SPAC, water potential decreases from the soil, soil, through the xylem, xyle, and the leaf apoplast at the evaporating surface of the substomatal cavity, SSC-APO, to the atmosphere, air, with soil greater than exyle, greater than SSC-APO greater than air, driving water flow to the atmosphere, while the stomatal resistance, 1/GS, sets the AE trade-off at the pore, as shown in Figure 2.1a, inset. The distribution of resistances and turgor mechanics from xylem supply to guard cell pressure in the outside xylem zone, or OXZ, that shapes the stomatal conductance response to varying environmental conditions, such as drought, which means decreasing exyle, and increasing vapor pressure deficit, or VPD, remains poorly understood. Despite decades of progress, the mechanistic basis of stomatal regulation under water stress remains unresolved. The debate concerns the relative contributions of hydropassive and hydroactive responses. In hydropassive regulation, water flows passively into or out of guard cells in response to local water potential changes, adjusting turgor and aperture without active osmotic regulation. In hydroactive regulation, underwater stress guard cells actively regulate osmolytes, primarily via the A-mediated pathway, enabling sustained stomatal closure under stress. In angiosperms, guard cells, abbreviated G, are flanked by subsidiary cells, abbreviated S, creating an epidermal mechanical advantage. A uniform decline in sesi across the leaf tends to open the stomatal pore rather than close it. When leaf water potential falls, subsidiary cells lose turgor and relax their mechanical constraint on guard cells, allowing the pore to open passively. Consequently, for drought-driven closure to occur, guard cell turgor must decline disproportionately relative to subsidiary cell turgor, a condition passive hydraulics alone struggles to produce. Buckley and colleagues showed this with a mathematical model and argued that hydroactive regulation is necessary for both xylem stress and VPD-driven steady-state stomatal closure. The hydropassive-hydroactive debate has therefore remained unresolved, not over whether hydroactive regulation is necessary, but over how much each contributes under varying stress. This question cannot be answered from wild-type gas exchange measurements alone, in which hydropassive and hydroactive mechanisms operate simultaneously and cannot be separated from the net stomatal conductance signal. The same leaf gas exchange measurements are also built on three coupled assumptions. Assumption A1. The intercellular airspace remains near saturation, with HSSC-APO approximately equal to 1. Assumption A2. Local thermodynamic equilibrium is maintained between symplasm and apoplasm during transpiration. And Assumption A3. Stomata present the dominant resistance to transpiration, meaning 1/GS is much greater than 1/GOXZ, where GOXZ, measured in millimoles per meter squared per second, is the conductance of the OXZ. Early experiments using inert gas tracing detected resistances in the water vapor pathway that were inconsistent with a purely stomatal limitation on water vapor transport, providing the first hints that the OXZ posed non-negligible resistance. More recently, oxygen-18 isotope discrimination combined with dual-sided gas exchange and inert gas diffusion provided indirect but increasingly compelling evidence that OXZ airspaces fall below saturation under moderate water stress and elevated VPD, and that symplasmic-apoplasmic disequilibrium develops well above the turgor loss point. These more recent findings collectively challenge assumptions A1 through A3. However, these techniques left open the alternative that apparent undersaturation could arise from stomatal patchiness or vascular flux partitioning artifacts in gas exchange calculations, without requiring a loss of GOXZ. This was resolved by Jane et al., who used aquadust, a nanoscale FR-ET-based hydrogel reporter injected into the leaf apoplasm to directly measure water status simultaneously upstream, that is CIXYL, and downstream, that is CISSC-APO, of the OXZ in intact transpiring leaves. Their core finding was an approximately 25-fold decline in GOXZ beginning well above the Turgor loss point, where CSI-TLP is approximately -2 megapascals, accompanied by strong OXZ apoplasmic undersaturation, with CISSC-APO approximately -10 megapascals, and cell-scale confocal evidence of extreme symplasmic apoplasmic disequilibrium in apparently turgid mesophyll cells. Critically, they observed that this OXZ conductance loss was entirely independent of VPD and of stomatal behavior, and it produced measurable gains in IWUE relative to predictions from GS alone. This VPD independence stands in direct contrast to the evaporative demand-driven undersaturation proposed by Marquez et al. And Wang et al., who attributed undersaturation in the OXZ primarily to high VPD rather than to declining CSI-XYL. These findings also undermine the vapor phase sensing framework of Peak and Mott, which requires the airspace to remain close to saturation. That is assumption A1 and has been incorporated into dynamic guard cell models such as OnGuard. Understanding how the hydraulic architecture of the OXZ, including loss of GOXZ, and the resulting undersaturation under water stress, shapes the water potential signal that guard cells experience, and how that drives stomatal regulation is therefore essential for mechanistic models of stomatal conductance. Two complementary advances, combined here for the first time, provide the experimental leverage needed to isolate the HP contribution. First, AquaDust directly resolves both CI-APOS-SC downstream and CSI-XIL upstream of the OXZ in intact, transpiring leaves of maize without any assumption of internal saturation. This simultaneous access to both ends of the OXZ enables GS and GOXZ to be computed with an undersaturation-corrected vapor driving force and provides a clean local measure of xylem water stress. Both capabilities are inaccessible to conventional gas exchange instruments and pressure chamber methods, which assume internal saturation and measure bulk leaf water potential, respectively. Second, the maize slack 1,2 null mutant W22 inbred background lacks functional ZMSL-AC1, the primary anion channel mediating guard cell anion efflux during ABA and stress-induced closure. As highlighted in the inset of Figure 2.1b, in wild-type plants, ZMSL-AC1 supports active turgor regulation, where psi equals P minus Pi, with functional ion channels, while in the SELAC1 mutant, the non-functional SLAC1, indicated by a red cross, blocks anion efflux and osmotic regulation, confining stomatal aperture changes to those driven by passive water redistribution alone. This genetic suppression of the HA mechanism, combined with AquaDust measurements that specify the hydraulic boundary conditions, Psi-XIL and Psi-SSC-APO, actually experienced by guard cells, allows the HP contribution to be quantified directly, providing the empirical foundation for informing a mechanistic model of stomatal conductance developed here. Here, we present the first direct comparison of stomatal and outer xylem physiology between maize wildtype and SLAC1 mutant plants across a progressive drought trajectory, combining steady-state gas exchange with AquaDust water, potential measurements to resolve GS, PSI-SSC-APO, GOXZ, and their ratio GS divided by GOXZ simultaneously in both genotypes, as shown in Figure 2.3. We use this dataset to confront a hydromechanical model of stomatal conductance and address an open question. What fraction of stomatal regulation in maize is explained by a purely hydropassive framework, as shown in Figure 2.4? The HP model is parameterized exclusively against SLAC1 data, in which HA regulation is genetically suppressed, and its ability to predict wild-type behavior is tested. Any failure quantifies where and how much HA input is required, as shown in Figure 2.5. The mechanistic insights generated here carry direct implications for crop improvement. Beyond stomatal physiology, the ratio GS divided by GOXs emerges as a physiological phenotype that captures whether a genotype exploits non-stomatal outer xylem resistance effectively. When large standard gas exchange instruments underestimate true GS due to apoplastic undersaturation, and intrinsic water use efficiency gains accrue from outer xylem resistance independently of stomatal closure. When small, no such advantage emerges. The quantitative model developed here connects cell-scale biophysical parameters to this leaf-scale phenotype, making GS divided by GOXC actionable as both a screening metric and a modeling target. Linking this phenotype to its genetic basis is a natural next step. Precision phenotyping of drought-related traits has driven sustained genetic gain in maize, and GS divided by GOXC represents an analogous mechanistically grounded target for future genomic dissection and crop improvement under water-limited conditions. Section 2.2 Materials and Methods. Section 2.2.1 Plant Material. Maize, Zea maize L, W22 inbred line plants, that is wildtype, or WT, and the SLAC 1-2 null mutant in the same W22 background were used in this study. The SLAC 1-2 line was provided by R.J. Tuhey III and A.J. Studer at the University of Illinois Urbana-Champaign. Plants were grown from seed, and measurements were conducted on the uppermost fully expanded leaf at the V9 through V10 vegetative growth stage. Section 2.2.2 Growth Conditions and Drought Treatment. Plants were grown in a greenhouse under controlled conditions. Day temperature 28 plus or minus 2 degrees Celsius, night temperature 20 plus or minus 2 degrees Celsius, 40% relative humidity during the day, and 80 to 90% at night, with natural sunlight supplemented to maintain a minimum photosynthetically active radiation, or PAR, of 1000 mu-mol M-2S-1 during a 16-hour photoperiod. Plants were grown in pots with potting medium containing slow-release fertilizer and watered daily until the start of the drought treatment. Drought stress was imposed by withholding water over 2 to 3 consecutive days, leading to a progressive decline in Sahil from well-watered conditions, where Sahil was approximately negative 0.2 megapascals, to near the turgor loss point, where TLP was approximately negative 2 megapascals. Measurements were conducted across this drought trajectory to sample a range of exile values independent of VPD. Section 2.2.3 Gas Exchange Measurements. Steady-state gas exchange was measured using a LI-Core 6800 portable photosynthesis system from LI-Core Biosciences, Lincoln, Nebraska, equipped with a standard 6 square centimeter leaf chamber. Measurements were performed on the adaxial surface of the uppermost fully expanded leaf at the V9 through V10 stage. Chamber conditions were maintained at a leaf temperature of 36 degrees C, a PR of 600 mu-mol per square meter per second, a CO2 concentration of 440 mu-mol per mol, and VPD varied between 2.5 and 5.0 kilopascals across measurements. VPD and exile were varied independently. The LI-Core VPD set point was decoupled from the plant's water status, allowing the same exile to be sampled under different VPDs and vice versa. A measurement was recorded as steady state when the coefficient of variation in transpiration rate, E, fell below 1% over a 3-minute averaging window. Measurements were performed between 0900 and 1500 to minimize diurnal drift. Section 2.2.4 In Planta Apoplastic Water Potential Measurements with Aquadust. Equation 2.1, Section 2.2.5 Stomatal Conductance Correction for Apoplastic Undersaturation. The standard gas exchange calculation of stomatal conductance, as implemented internally by the LI-Core 6800, treats the leaf intercellular airspace as saturated with water vapor at the leaf temperature. Aquadust measurements made directly in the apoplast show that this assumption is violated in transpiring leaves as exile decreases. We corrected each Li core-based GS reading using the AquaDust PSISSC-APO measured concurrently on the same leaf. Throughout the study, GS refers to this AquaDust corrected value. Combining Li core gas exchange with concurrent AquaDust water potential readings is what makes this correction possible. Neither instrument can produce it alone. Equation 2.2, where Wi equals WW subscript S SCAPO, measured by AquaDust, replaces the standard assumption of saturation at leaf temperature. Section 2.2.6, Hydromechanical Model and Bayesian Inference. To isolate hydraulic contributions to stomatal regulation, we fit a steady-state three-compartment hydromechanical model to the SILAC-1 dataset by Bayesian inference. The model represents the leaf as mesophyll, subsidiary cell, and guard cell compartments connected in series from the xylem boundary, with stomatal conductance proportional to the guard cell subsidiary cell turgor difference. The outside xylem conductance, GOXZ, as a function of CIXYL, calibrated from concurrent AquaDust measurements, was supplied as a known input. Full model equations are given in SY section A3. Prior distributions for all 12 free parameters are given in SI sections A4 and A5.4, and MC convergence diagnostics are given in SI section A5.6. Parameter estimation used the EMC ensemble sampler. The posterior was then used to generate forward predictions for wildtype GS and PSI ApoSSA. Systematic deviation of wildtype data from these HP model predictions is interpreted as evidence for active hydroactive, or HA, regulation. Section 2.2.7, Software and Code Availability. All analysis and inference were performed in Python, version greater than or equal to 3.10, using NumPy, SciPy, Pandas, Matplotlib, and MC. Steady-state solutions of the three-compartment model were obtained with the SciPy.optimize route finder. Analysis code, run configurations, and prior definitions are available at https://github.com/desai-sahil/maize-hydraulics. Section 2.3.1, Comparison of physiological response between wildtype and SALACT1 stomatal mutant in maize. To interrogate the relative contributions of stomatal and non-stomatal regulation, we compared the hydraulic and gas exchange responses of wildtype and the SALAC1 null mutant in the W22 background across a progressive drought trajectory parameterized by CSI-XIL, as shown in Figure 2.3. We measured stomatal conductance, referred to as GS, apoplastic water potential in the substomatal cavity, referred to as PSISSCAPO, outside xylem zone conductance, referred to as GOXZ, and their derived ratios as a function of xylem water potential in both genotypes, again as shown in Figure 2.3. Across both genotypes, GS and GOXZs showed weak dependence on VPD relative to their dependence on CSI-XIL, as shown in supplementary Figure A1. Measurements collected across the VPD range of 2.5 to 5.0 kilopascals were therefore pooled within each CSI-XIL bin, with each plant contributing at most one biological replicate per bin, as described in the methods. Stomatal conductance, that is, GS. Under well-watered conditions, where CSI-XIL equals negative 0.2 megapascals and negative 0.6 megapascals, SLAC1 exhibited significantly higher GS than wild type. As xylem potential declined towards moderate drought stress, where CSI-XIL equals negative 1 megapascal and negative 1.4 megapascals, GS in the SLAC1 mutant declined more sharply compared to wild type, which remained relatively stable and unchanged from well-watered to moderate stress. At extreme drought stress, where CSI-XIL equals negative 1.9 megapascals near turgor loss, GS declined significantly for both genotypes, with the wild type and SLAC1 converging to similar values. Outside xylem water potential, CISSC-APO. Despite these pronounced differences in stomatal conductance, the apoplast water potential at the substomatal cavity, CISSC-APO, did not differ significantly between the wild type and SALAC1. With increasing water stress, CSI-SSC UPO initially declined before recovering at high water stress as stomata closed. However, the potential did not return to its pre-stress, well-watered state. Notably, both the wild-type and CELAC1 converged in water potential at moderate to high water stress levels. Outside xylem conductance, GOX declined sharply and similarly in both genotypes with decreasing xylem water potential. There was no statistically significant difference between the wild-type and CELAC1 at any stress bin, confirming that the loss of outside xylem conductance is driven predominantly by xylem potential and is entirely independent of stomatal behavior. Phenotype GS/GOXZ. By evaluating the ratio of stomatal to OXZ conductance, written as GS/GOXZ, we quantified this shifting control of transpiration. While the GS/GOXZ ratio remained low in well-watered conditions, indicating stomatal dominance, it increased significantly as stress progressed. Within the CELAC1 mutant, the ratio GS/GOXZ increased significantly from well-watered conditions, peaking at PSI-XYL equals -1 megapascal and -1.4 megapascals before declining at severe stress, where PSI-XYL equals -1.9 megapascals, as stomatal closure reduced transpiration flux, as shown in Figure 2.3d. Wild-type showed qualitative and quantitative trends similar to SLAC1. Finally, we assessed the impact of this non-stomatal limitation on standard gas exchange calculations. The fractional error in estimating stomatal conductance, calculated as GS minus GS, all over GS, with GS being the conventional value obtained under the saturated airspace assumption, increased significantly and linearly with the GS/GOXZ ratio across both genotypes, as shown in Figure 2.3e, reaching approximately 20 to 25% at the highest observed ratios. This strong correlation demonstrates that as the OXZ pathway's resistance becomes comparable to stomatal resistance, conventional measurements that assume internal saturation will substantially miscalculate actual stomatal conductance. Section 2.3.2 Hydropassive HP Model Predictions and Parameter Fitting. Section 2.3.3 Model Validation with Experimental CELAC1 Data. To characterize the localized water transport pathways from the xylem to the substomatal cavity and to the atmosphere, we formulated a resistance capacitance, or RC, hydropassive model that represents the hydraulic conductances, resistances, and local water potentials of the mesophyll, subsidiary, and guard cell compartments, as shown in Figure 2.2b. Because steady-state measurements of GS and CSI-SSC APO alone cannot uniquely determine all 12 free biophysical parameters, we used Markov chain Monte Carlo, or MCMC, Bayesian inference to explore the parameter space and quantify posterior distributions for each parameter. To parameterize the model, we used the experimentally determined vulnerability of the outside xylem conductance, GOXZ, as a known input, shown in Figure 2.3c. We then evaluated the predictive performance of the HP model across the SLAC1 dataset by comparing model predicted values against all individual steady-state observations of GS and SSI APO-SSC. The observed versus predicted comparison aligned along the 1-to-1 reference line for both observables, with R squared equal to 0.52 for GS and R squared equal to 0.84 for PSI APO-SSC, as shown in Supplementary Information Figure A5. In the main text, Figure 2.4 shows the corresponding observed and model predicted distributions binned by xylem water potential. To visualize the trajectory of the model response across the drought gradient, we evaluated the fitted HP model at the same PSI-XYL, VPD, and GOXZ inputs as in the SLAC1 experiments, generating a predictive distribution for each steady-state observation. The resulting per-plant predictions were binned by PSI-XYL, in the same manner as the data to produce box plots of predicted plant medians plotted alongside the experimental medians, as shown in Figure 2.4. Across bins, the median model prediction closely tracked the empirical medians, reproducing the gradual, near-linear decline of GS from well-watered to stressed conditions, as shown in Figure 2.4a. The model also captured the strongly nonlinear trajectory of Psi-SSC-APO, including the steep drop to approximately negative 13 megapascals at moderate stress, where Psi-XIL was approximately negative 1 megapascal, and the partial recovery of Psi-SSC-APO with stomatal closure at extreme stress, as shown in Figure 2.4b. The broader posterior predictive intervals on Psi-SSC-APO compared to those on GS arise because the model propagates the high uncertainty of internal undersaturation to per-plant variation in Psi-XYL. The higher R-squared for Psi-SSC-APO, 0.84, versus 0.52 for GS, reflects the model's ability to track this strong Psi-XYL dependence, while the lower R-squared for GS indicates residual biological scatter in stomatal conductance beyond what the purely hydropassive framework captures. The drop in Psi-SSC-APO at moderate stress arises as GOXZ declines with decreasing Psi-XYL, increasing the water potential drop across the outside xylem pathway. At more severe stress, stomatal closure reduces evaporative flux and partially relaxes this drop, leading to recovery in Psi-SSC-APO. The model reproduces both phases of this response without being tuned to the shape of the trajectory, indicating that these trends emerge directly from the hydraulics. Taken together, these results show that a purely hydropassive framework, assigning no role to ABA-mediated active osmotic regulation in the guard cells, is sufficient to quantitatively explain both stomatal conductance and internal water status in the SILAC1 mutant across the full drought trajectory. A table is shown here. References and derivation of physiological ranges are provided in Appendix A, Section A4. A note for the listener. This thesis refers to appendix material, which is not included in the audiobook. You can find it in the full written text. Section 2.3.4, Parameter Identifiability and Physical Insights. Analysis of the posterior distributions from the MC-MC Bayesian parameter estimation provides critical insight into which biophysical parameters govern the steady-state physiology of the SLAC1 mutant. All 12 posterior distributions were unimodal. To quantify the sharpness of each posterior, we computed a relative uncertainty defined as half the width of the 95% credible interval divided by the posterior median, shown in Table 2.1. Parameters fell into two clear groups, those with relative uncertainties below 1, meaning tightly constrained, and those above 1, meaning weakly constrained. The group comprising the maximum guard cell and subsidiary cell turgor pressures, PG0 and PS0, the cell elastic moduli, epsilon G and epsilon S, the turgor conductance scaling factor, Chi, and the cuticular conductances to the atmosphere, GG2 and GE squared, is tightly constrained. In the model, stomatal conductance is proportional to the turgor pressure difference between guard and subsidiary cells. GS equals chi times the maximum of 0 and PG minus M times PS, where M is the guard subsidiary mechanical advantage, with M equal to 1.71, as given in SAI Table A1. The observed GS therefore directly and sensitively encodes the product chi times the quantity PG minus M times PS, making chi, PG0, and PS0 the primary determinants of the observable. Similarly, the elastic moduli govern how turgor responds to changes in cell water content, expressed as delta P equals epsilon times delta 5 divided by V, controlling how sensitively PG and PS track Psi-XIL along the drought trajectory. The curvature of the GS versus Psi-XIL relationship therefore encodes both epsilon values. The cuticular conductances GG squared and GE squared are likewise tightly constrained because they couple a large vapor pressure deficit between the guard subsidiary cells and the atmosphere. Even modest changes in Gg squared and Ge squared therefore produce appreciable shifts in guard and subsidiary cell water potential, altering turgor and, through chi times, the quantity Pg minus m times Ps, stomatal conductance. The narrow posteriors, with relative uncertainties of 0.05 to 0.55, as shown in Table 2.1, confirm that the paired dataset of GS and PSIS-SC-APO provides sufficient information to resolve these parameters without ambiguity, and the inferred values fall within ranges established by direct cell mechanical measurements. The second group, comprising the mesophyll to subsidiary hydraulic resistance, RMS, the subsidiary to guard cell resistance, RSG, the internal vapor conductances to the SSC, GG1, and GE1, and the maximum guard cell volume, VG0, is weakly constrained. The internal vapor conductances GG1 and GE1 from the guard and subsidiary cells are insensitive at steady state because they are multiplied by relatively small vapor pressure differences between the cell interiors and the SSC. Multiple combinations of GG1 and GE1 therefore produce equally good fits, yielding wide posteriors with relative uncertainties of 1.92 and 3.73. Similarly, the series hydraulic resistances RMS and RSG are weakly constrained because steady-state data cannot independently apportion the total xylem to guard cell pressure drop between the two series elements. Notably, despite its wide posterior, RMS strongly prefers low values, with a posterior median of 0.032 megapascals times meters squared times seconds per millimole, indicating a dominance of liquid phase transport over vapor phase conductance from the mesophyll to epidermis. Finally, the guard cell volume VG0, with a relative uncertainty of 1.09, is borderline weakly constrained. At steady state, it affects model outputs primarily through the osmotic concentration, already partially constrained by Pg0 and epsilon g. Resolving Vg0 independently would require direct volumetric measurements by confocal microscopy of the stomatal complex in maize. From an experimental design perspective, the identifiability analysis shows a clear priority hierarchy for future measurements. Maximum turgor pressures, Pg0 and PS0, can be estimated via pressure probe micromanipulation of individual guard and subsidiary cells in epidermal peels, or, for bulk averages, from pressure volume curve analysis. The elastic moduli epsilon g and epsilon s can be derived from the turgor versus relative water content relationship in pressure volume curves, or more directly from concurrent pressure probe and confocal volume measurements, who reported guard cell elastic moduli of approximately 2 to 40 megapascals in Vicia faba, consistent with our inferred epsilon g equals 3.11 megapascals, as shown in Table 2.1. The scaling factor chi is accessible through combined measurements of stomatal aperture, guard and subsidiary cell turgor, and leaf stomatal geometry. Section 2.3.5, Parameter Correlations and Biological Coupling. To determine whether weakly constrained parameters were mathematically compensating for one another, we analyzed the pairwise parameter correlation, shown in supplementary figure A4. Several strong correlations emerged, and their interpretation reveals that the MCMC sampler respects both the physical and geometric constraints of the model, producing correlated posterior solutions that reflect either genuine anatomical scaling or mathematical coupling through the model. Strong positive correlations were observed between the guard and subsidiary cell elastic moduli, epsilon g and epsilon s, with R equals 0.87, and between the maximum turgor pressures, Pg0 and PS0, with R equals 0.80. These correlations merit careful interpretation. Because GS is driven by the turgor pressure difference, Pg minus m times PS, the observable GS primarily constrains the difference between guard and subsidiary cell parameters, rather than each absolute value independently. The MCMC sampler therefore explores the posterior landscape of Pg0 and PS0 together, along which epsilon g and epsilon s covary to maintain the same net turgor sensitivity. This is principally a mathematical coupling arising from the structure of the GS equation, rather than a direct biological linkage. A second class of correlations reflects geometric scaling imposed by cell anatomy. The subsidiary cell volume VS0 is set equal to VG0 in our model, and as a result, the cuticular conductance GE squared correlated strongly with VG0, with R equal to 0.76. Larger cells expose greater surface area, necessitating proportionally higher vapor losses to maintain the steady-state water potential gradient. Similarly, the subsidiary to guard cell hydraulic resistance, RSG, exhibited a negative correlation with VG0, with R equal to negative 0.52, because resistance scales inversely with cross-sectional area. These correlations therefore faithfully mirror anatomical scaling laws and represent geometric coupling, rather than fitting artifacts, indicating that the MCMC sampler recovers internally self-consistent cell geometries. Importantly, several pairs of weakly constrained parameters showed near-zero posterior correlations, providing a critical mechanistic clarification. The internal vapor conductances GG1 and GE1 were essentially uncorrelated, with R equal to 0.04, demonstrating they do not compensate for one another, despite both being weakly constrained. This confirms that their wide posteriors reflect genuine insensitivity of the steady-state outputs to each parameter individually, not a mathematical trade-off between them. Likewise, the series resistances RMS and RSG were uncorrelated, with R equal to 0.08, demonstrating that the model can mathematically distinguish upstream from downstream resistances even when their absolute magnitudes remain loosely bounded by the data. The absence of compensatory correlations among the weakly constrained parameters confirms that their limited identifiability reflects the information content of the steady-state observables. Section 2.3.6 Hydropassive Model Fit to Wild-type Data. Having established that a purely hydropassive, or HP, framework quantitatively captures both stomatal conductance and internal water status in the CELAC1 mutant across the full drought trajectory, we next asked whether the same framework could account for wild-type, or WT, physiology. To test this, we applied the identical MCMC Bayesian inference procedure to the WT dataset, fitting the HP model independently to WT measurements of GS and PSI-SSCAPO, with the WT-specific GOXZ of PSI-XYL vulnerability curve supplied as a known input, as shown in Figure 2.3c WT. This procedure is a direct test of whether a purely passive hydraulic framework, with no active osmotic regulation in the guard cells, is sufficient to explain the GES observed in WT plants. For GS, the model fit was substantially weaker compared to SLAC1, with an R-squared of 0.29 versus 0.52 for SLAC1, as shown in Figure 2.5a. Across bins, the predicted medians overestimated observed wild-type GS at well-watered and extreme water stress conditions, where psi-XIL equals negative 0.20 and negative 1.90 megapascals, and underpredicted versus data at moderate stress, where psi-XIL equals negative 0.6, negative 1, and negative 1.4 megapascals. In contrast, the HP model reproduced the wild-type psi-S SCAPO trajectory with accuracy comparable to that achieved in SLAC1, with an R-squared of 0.78 versus 0.84, as shown in Figure 2.5b, and in supplementary Figure A6 and supplementary Table A6. The predicted binwise distributions of PSI-SSCAPO closely tracked the observed medians across all psi-XIL bins, including the steep drop at moderate stress and the partial recovery at extreme stress. Because GOXZ is genotype invariant and supplied as a measured input rather than a free parameter, this result confirms that the hydraulic architecture of the outside xylem zone is shared between the two genotypes and is captured by the model in both. The model failure is therefore localized specifically to GS. To characterize the nature of this failure more precisely, we computed the per-plant residuals delta GS equal to GS predicted minus GS observed, and plotted these against Sahil as shown in Figure 2.5c. The residuals are scattered approximately symmetrically around zero across the full extent of water stress, with large positive and negative deviations present at every Sahil bin. There is no systematic directional bias. In particular, the HP model does not consistently over or underpredict WTGS. This rules out a simple, uniform HA-driven suppression of GS as the primary explanation for the model shortfall. Comparing the MC-MC inferred parameter sets between genotypes reveals the mechanistic cost of fitting WT within a purely HP framework, as shown in Table 2.1 and Supplementary Information Table A5. The tightly constrained cell mechanical parameters are nearly identical between genotypes. Turgor pressures, Pg0 at 2.49 versus 2.50 megapascals, and PS0 at 0.97 versus 1.04 megapascals. Elastic moduli, epsilon G at 2.81 versus 3.11 megapascals, and epsilon S at 0.29 versus 0.25 megapascals. And cuticular conductances, GG squared and GE squared, all agree closely. This agreement is expected, as both genotypes share the same W22 inbred background. The fundamental cell mechanical properties of the stomatal complex should not differ between them. The Turgor conductance scaling factor Chi is modestly lower in WT at 367 versus 439 millimoles per meter squared per second per megapascal, and the maximum guard cell volume VG0 is smaller at 205 versus 334 millimoles per meter squared of leaf area. These differences likely reflect minor morphological variation in stomatal complex geometry between the two lines. The liquid pathway hydraulic resistances, by contrast, differ dramatically between genotypes. The mesophil to subsidiary resistance, RMS, is 24-fold higher in wild type, 0.776 versus 0.032 megapascals times square meters times seconds per millimole. The subsidiary to guard cell resistance, RSG, is 10-fold higher, 2.784 versus 0.27 megapascals times square meters times seconds per millimole. Both values remain within the physiological bounds, but are 1 to 2 orders of magnitude above the SLAC1 inferred values. Within the HP model, elevated liquid pathway resistance limits water delivery to the guard cell relative to the subsidiary cell, causing PG to decline more steeply than PS as SIXYL falls, and thereby reducing GS. The MC-MC finds this high-resistance configuration as the only HP consistent explanation for the lower GS of wild type. The subsidiary cell vapor conductance GE1 is also approximately 10-fold higher in wild type, 24.31 versus 2.35 millimoles per square meter per second. This is a weakly constrained parameter in both fits, whose elevated value in wild type may reflect partial compensation for the high liquid pathway resistances through increased vapor phase flux to the SSC. The most parsimonious interpretation of the elevated RMS and RSG in wild type is that these parameters serve as a model surrogate for the active osmotic turgor reduction that ZMSLAC1 mediated anion efflux imposes on guard cells. In wild type, ABA-mediated anion efflux through SLAC1 actively reduces guard cell osmolyte concentration, lowering πG and hence PG below what passive water redistribution alone would predict. Because the HP model has no mechanism to represent this active turgor reduction, the MC-MC compensates by inflating the liquid pathway resistances, effectively mimicking the turgor deficit through a structural parameter. The scatter-dominated residuals in Figure 2.5c arise because, with RMS and RSG fixed as constants, the HP model predicts essentially the same GS for any two plants at the same SIXYL. In wild type, however, the actual GS at a given CIXYL depends not only on hydraulic state, but also on the ABA signaling status of each plant, which may be subject to biological variability in biosynthesis, transport, and guard cell sensitivity that cannot be reduced to a single CIXYL value. The genotype comparison in Figure 2.6 reinforces this conclusion. In the well-watered stress range, where wild-type and SLAC1 GS differ significantly, as shown in Figure 2.3a with different significance letters, the HP model predicts lower GS for wild-type than for SLAC1. This is consistent in direction with the observed difference, but the predicted wild-type distributions do not fully reach the low values observed experimentally, as shown in Figure 2.6a. Simultaneously, the HP predicted psi-S-SC-APO distributions for wild-type and SLAC1 are statistically indistinguishable across all stress bins, as shown in Figure 2.6b, consistent with the experimental finding that psi-S-SC-APO does not differ significantly between genotypes at any psi-XYL level, as shown in Figure 2.3b. This dissociation, where the model fails specifically on GS while succeeding on psi-SSC-APO, makes a case for an active regulatory component, genetically suppressed in SLAC1 by the loss of functional ZIA-MSLAC1 that is required to fully account for wild-type stomatal physiology. We combined direct implanta measurements of apoplastic water potential with a stomatal mutant that removes active anion efflux from guard cells to separate passive and active contributions to stomatal regulation in maize. A purely hydropassive model, parameterized entirely on SLAC1 data, reproduced both stomatal conductance with an R² of 0.52 and apoplastic water potential at the substomatal cavity, which we call CISSC-APO, with an R² of 0.84 across the full drought trajectory, including the steep decline and partial recovery of CISSC-APO. The same model, applied independently to wild-type, provided a weaker fit to stomatal conductance with an R² of 0.29, while still predicting psi-SSC-APO with comparable accuracy, with an R² of 0.78. This failure on GS alongside the accurate prediction of psi-SSC-APO points to missing regulation in guard cell osmotic control, rather than in the hydraulic pathway that sets outside xylem water status. Steady-state closure requires guard cell turgor to fall disproportionately relative to subsidiary cell turgor due to the subsidiary cell mechanical advantage, and passive water redistribution alone struggles to capture the wild-type stomatal response. In our model, the conductance relation GS proportional to chi times, open parenthesis, PG minus MPS, close parenthesis, is a linearized representation of this mechanical advantage. To fit wild-type within a purely passive framework, the MCMC inference must inflate the liquid pathway resistances, raising RMS roughly 24-fold and RSG roughly 10-fold relative to CELAC1, while leaving the tightly constrained cell mechanical parameters essentially unchanged. We interpret this not as a genuine anatomical difference, which a single gene knockout would be unlikely to produce, but as a structural surrogate for the osmotic turgor reduction that ZMSLAC1, mediated anion efflux, imposes on guard cells in wild-type. Outside xylem conductance declines with xylem water potential in both genotypes and is independent of stomatal behavior, extending the non-stomatal control of transpiration recently described in maize. Because G outside xylem does not differ between wild-type and SLAC1, the conductance loss sits upstream of the guard cells, and the accompanying drop of psi-SESC-APO to roughly negative 13 megapascals places the substomatal apoplast far below the bulk turgor loss point. Undersaturation has been reported to track vapor pressure deficit, while our G outside xylem responds to xylem potential with no detectable VPD dependence. These findings carry direct consequences for how stomatal conductance is measured under drought. When computed under the standard assumption of a saturated intercellular airspace, GS is underestimated by 20 to 25% in our most affected plants, with the fractional error increasing approximately linearly with the ratio of GS to GOXZ. Because this bias is largest under moderate stress, it propagates into estimates of intrinsic water use efficiency and into the leaf-scale relationships on which larger-scale models rely. Our conclusions are bounded by a steady-state model, a single SLAC1 allele evaluated under greenhouse conditions, and inferred rather than directly measured guard cell mechanics. Direct measurements of guard cell turgor and volume, and additional alleles and field environments would test how far the passive-active partition generalizes. The present data nonetheless establish a firm baseline. In maize, a hydropassive model is sufficient once active AIN efflux is genetically removed, and the residual that reappears in wild type marks the ZMSLAC1 dependent contribution of active osmotic regulation. Beyond mechanism, these results have implications for improving maize under water-limited conditions. Sustained genetic gain under drought has largely come from selection on physiological traits and from managed stress phenotyping pipelines that quantify how plants acquire and conserve water. Our work identifies an axis that these pipelines do not currently resolve: outside xylem hydraulic vulnerability, described by GOXZ and by the composite ratio GS divided by GOXZ. Because GOXZ declines with xylem water potential independently of stomatal behavior and does not differ between wild type and SLAC1, it defines a control point upstream of stomata that conventional gas exchange systematically misreads under stress. A phenotyping strategy that measures this internal decline could reveal heritable differences in how efficiently water is delivered to evaporating surfaces and would complement the stomatal and canopy traits that maize drought improvement has so far emphasized. Chapter 3. Abscisic acid-mediated water stress regulation can mechanistically explain oscillations and water stress memory in stomatal conductance. S. Desai and A.D. Stroke, 2026. Abscisic acid-mediated water stress regulation can mechanistically explain oscillations and water stress memory in stomatal conductance. BioArchive. DOE 10.64898/2025.12.26.696581. Author contribution: S. Desai and A.D. Stroke designed the research. S. Desai developed the mathematical model. S. Desai and A.D. Stroke analyzed the results and wrote the paper. Section 3.1 Summary. Stomatal pores, formed by guard cells, govern the critical trade-off between carbon assimilation and water loss in plants. Their dynamic responses to environmental stresses, such as stomatal oscillations and drought stress memory, also called hysteresis, have lacked a unified mechanistic explanation. While abscisic acid, or ABA, is believed to play key roles in water stress responses, no model has linked its core regulatory kinetics to these complex stomatal behaviors. Here, we introduce a coupled hydropassive hydroactive model, the HP-HA model, that integrates leaf hydraulics with the biokinetics of guard cell autonomous ABA regulation and plasma membrane-mediated osmoregulation. We demonstrate that this framework predicts accurate, genotype-specific stomatal regulation across wild type, ABA-insensitive mutant, OST1-3, and ABA synthesis mutant, AAO3-2, in Arabidopsis thaliana, abbreviated AT, and that nonlinear feedbacks in ABA autoregulation can drive both stomatal oscillations and hysteresis. This work provides a unifying mechanistic framework linking ABA autoregulation and signaling to hydropassive leaf hydraulics that reproduces oscillations and water stress memory in stomatal conductance, offering a foundation for understanding plant responses to water stress. Section 3.3 Methods. This section provides a concise overview of the hydropassive and hydroactive model formulations, detailed derivations for both models, a complete list of parameters, and the solution approach are presented in the supplementary information, sections B2 through B4 and B7, section 3.3.1, Hydropassive Model. The HP model describes the regulation of stomatal aperture via changes in guard cell volume and turgor-driven, primarily by hydraulic signals. Stomatal conductance is determined by guard cell turgor pressure and the back pressure from subsidiary cells. At the leaf scale, passive water movement through the xylem, mesophyll, subsidiary cells, and guard cells affects guard cell turgor pressure, which in turn influences stomatal conductance. These physical processes in the HP model are represented using an electrical analogy of hydraulic capacitances, resistances, and water fluxes, driven by differences in water potential. The model formulation is provided in equations B1 through B39 and parameterized using values in tables B4 through B5. Section 3.3.2. Hydroactive model. Section 3.3.3. Coupling eighths. The HP and HA models are coupled at two key points. First, linking water status to ABA biosynthesis. Changes in guard cell turgor pressure, driven by water status in the hydropassive model, trigger ABA biosynthesis, V stress, delta PG. This represents water stress stimuli from the hydropassive model informing a biochemical process in the hydroactive model. Second, feedback on turgor pressure. Changes in guard cell osmotic potential, governed by the hydroactive model, feedback to regulate turgor pressure, delta PIG. Changes in the guard cell osmotic potential then modify the cell's total water potential and guard cell turgor. This physiological osmotic adjustment thus affects stomatal conductance, completing the feedback loop between the two models. Section 3.3.4. Model parameterization. The HP model was parameterized to reproduce the stomatal response of the ABA-insensitive OST1 mutant. These parameters were then held constant for all genotypes. The HA model required additional parameters for guard cell electrophysiology, ABA signaling, and ABA regulation. Electrophysiology parameters were sourced from literature on AT, supplemented with data from Vitia Faba where necessary. ABA signaling rate parameters were taken directly from the literature. 6 unknown parameters for the ABA regulation motif: V stress, V plus, V minus, K plus, K minus, and theta sin were calibrated by matching model predictions to 6 experimental measurements from Marillo, creating a fully constrained system. Temperature enters the model only through the definitions of VPD, from relative humidity, and osmotic potential, from solute concentration. The chemical rate processes carry no explicit temperature dependence. We set T equal to 36 degrees C to match the VPD values of 1.1 and 2.2 kilopascals, corresponding to the relative humidities of 80% and 60% in the Marillo calibration dataset. Using a non-dimensionalized formulation of the ABA equations, described in SFLWine section B 5.2, we isolated the relative timescales of ABA and hydraulic dynamics. We employed a sequential fitting strategy. Wildtype parameters were determined by fixing theta sin equal to 1, constraining characteristic concentration ratios lambda minus and lambda plus, and fitting both transient and steady-state responses using the dimensionless groups omega-1, omega-2, and omega-3. Parameter values were selected from regions that reproduced the experimentally observed damped oscillations. The ABA synthesis mutant was then parameterized by varying theta sin while keeping other parameters fixed. Full details of the non-dimensionalization, fitting procedure, and bifurcation analysis are provided in SFAS section B5. Comprehensive tables listing all model parameters, their sources, and the methodology for parameter fitting are provided in SI section B7. Section 3.3.5. Numerical implementation. Full details of the numerical implementation, including the SciPy-Solve_IVP-BDF solver configuration, tolerances, initial conditions, and simulation protocols for each main text figure, are provided in SY section B7. That section also documents the 3 interactive Jupyter Notebook dashboards that allow readers to perturb any parameter and observe its effect on GS and ABA concentration in the guard cell in real time, along with the parameters and their plus or minus 10% steady state and dynamic sensitivity coefficients tabulated in SI tables B4 through B11. We parametrized our model by confronting its predictions with measurements reported by Merillo of stomatal response to a step change in vapor pressure deficit in 3 genetic variants of Arabidopsis thaliana. Arabidopsis thaliana wildtype, color 0, referred to as WT, and shown with green circles and curve, and Arabidopsis thaliana ABA signaling mutant OST1-3, referred to as SIG, and shown with blue triangles and curve, and an Arabidopsis thaliana ABA synthesis mutant AIO3-2, referred to as SYN, and shown with red diamonds and curve, all is shown in Figure 3.2b. Section 3.4.1, Hydropassive Framework can explain the features of an ABA signaling mutant. For the HP portion of our model, we modified the framework of Buckley to make the calculations of evaporative fluxes from mesophyll, EM, subsidiary cell, ES1 and ES squared, and guard cell, EG1 and EG squared, explicit, and to allow for internal undersaturation, where HSSC is less than 1, as shown in Figure 3.1d and described in the supplementary information. Assuming the responses of the signaling mutant, shown in Figure 3.2 a middle and Figure 3.2b as blue triangles, were dominated by hydropassive processes, we used this transient to adjust hydraulic parameters, hydraulic resistances, capacitances, osmotic potentials, evaporative resistances, and mechanics of guard and subsidiary cells, as depicted in Figure 3.1d, within ranges defined by the literature. Setting the total concentration of OST1 protein 3 orders of magnitude lower than the wild-type concentration, with OST1 total signaling equal to 1 nanomolar versus OST1 total wild-type equal to 1 mu-molar, in our HA biokinetic model shown in Figure 3.2 a middle. The predicted HP-dominated responses shown in Figure 3.2b as the blue curve agree within uncertainty, with the measurements shown as blue triangles in Figure 3.2b. Notably, the predictions capture high initial steady-state values of GS at times less than 0, and a weak loss of steady-state conductance in response to increasing water demand, where GS at times less than or equal to 0 minutes is approximately 300, and GS at t equals 60 minutes is approximately 280. They also capture a short-lived wrong-way response immediately after the imposed change in vapor pressure deficit that arises due to the opposing effect of subsidiary cell turgor, PS, and guard cell turgor, PG, on stomatal aperture, and the phase lag between these pressures, since PS responds before PG, as observed previously. We conclude, as did Buckley, that HP processes can explain the characteristic wrong-way response observed in angiosperms, but cannot explain the magnitude or details, for example, damped oscillations, of responses to change in evaporative demand. Section 3.4.2, ABA dynamics control the stomatal response in hydroactive genotypes. Using the HP parameters established above, we next examined predictions of the guard cell localized ABA-mediated HA processes in our coupled HPHA framework for wild-type responses and for a synthesis mutant. Key details of the HA model are provided in Methods and SI Section B3. We identify the rate processes of ABA. Autoregulation as the key control points for both biochemical and stomatal responses. The dynamics are governed by the interplay of, first, synthesis driven by stress, V stress. Second, positive feedback of ABA on its own synthesis, V+. And third, negative feedback of ABA on itself via A8H, V-. Importantly, the parameters V- and the ratio V+ divided by V stress govern whether the model predicts qualitative features such as damped or sustained oscillations and hysteresis in stomatal conductance. To select parameter values, We confront the predictions of this model of ABA dynamics with transient measurements of GS from Merillo for wildtype, abbreviated WT, shown as green circles, and an AAO3-2 mutant, abbreviated SYN, shown as red diamonds. We model the AAO3-2 mutant as having a weaker total synthesis rate, indicated by the red cross, and weaker cooperativity in the ABA biosynthetic pathway relative to wildtype, with all other parameters the same between the two. The weaker synthesis rate is indicated by theta SYN in equation B53, which modulates rates of ABA synthesis by both stress, V stress, and positive autoregulation, V+. We present predictions for a set of parameters that provide quantitative agreement with steady-state conductance for both genotypes, that is, the green and red curves for GS at times t less than or equal to 0 minutes and GS at time t equals 60 minutes, and qualitative agreement with features of the transients, particularly the damped oscillations in the wild-type plants. Notably, based on comparisons with this data and due to the nonlinearity of the governing equations, we cannot exclude other parameter values. In SLI figure B11, we present satisfactory predictions with an alternative set of parameters. As we will discuss further below, we identify the parameterization used in figure 3.2 as preferred based on the predictions it yields with stronger evaporative demand. To gain mechanistic insight into the predicted dynamics, we plot the trajectories of turgor pressure, Pg, ABA concentration, bracket ABA bracket G, activated fraction of slack1, and change in osmotic potential, delta PIG, in the guard cells. Focusing on wild-type dynamics, shown as green curves, we see the following progression. First, VPD acts through HP processes to lower Pg. Second, the drop in Pg initiates synthesis of ABA. Third, rising concentration of ABA drives its own degradation such that it quickly reaches a maximum, marked with a dashed green vertical line, and begins to fall. The observed fourfold change in guard cell ABA levels in the nanomolar range aligns with estimates reported in literature. Fourth, the time for activated slack1 and osmotic potential to rise to their maximum matches that for bracket ABA bracket G, as does the time to the first minimum in GS. We conclude that the rate and feedback encoded in ABA autoregulation plays the dominant role in defining the HA process in this model, with ABA signaling to CLAC1 and membrane-mediated osmoregulation both occurring rapidly relative to ABA synthesis and degradation. This predicted damped oscillatory response is consistent with gas exchange measurements following step changes in humidity. While damping and oscillations can be a signature of ABA-mediated responses, our ABA model predicts that such responses can also occur without oscillations, a result consistent with other experimental observations. The role of V- in controlling oscillations is explored in figure B9. Additional exploration of the speed of stomatal closure and comparison with experiments from Merillo, along with the important origins of the predicted dynamics, can be found in S1 section B 5.5 and figure B10. Section 3.4.3, oscillations in stomatal conductance emerge from ABA autoregulatory motif, AITS. In figure 3.3, we follow previous modeling efforts in using the qualitative change in stomatal dynamics from damped to persistent oscillations to identify a source of nonlinearity in the governing processes. Cowan presented an HP model in which the only nonlinearity came from the coupled heat and mass transfer from the leaf's interior to atmosphere. In contrast, Peake and Mott presented multi-stomate models in which they include nonlinear, hydropassive coupling between guard cells and light and CO2-dependent processes in individual guard cells. They predict the damped oscillation in response to a step increase in VPD and oscillations after a transition from white to red light. They did not report a transition from damped to persistent oscillations for a single type of perturbation. Neither of these studies provided a mechanistic explanation for this experimentally observed phenomenon, shown in figure B14. Instead, They only described the transition qualitatively, using simplified hydraulic representations with parameter estimates disconnected from the underlying biological processes. Here, we use our coupled HA model, parameterized to match the dynamics of wildtype AT, shown in Figure 3.2b, to explore the transient responses of stomata to successive step increases in VPD of equal size, as shown in Figure 3.3. Figure B15 presents an experimental example of this type of multi-step transition. After a first-step increase in vapor pressure deficit from 1.1 kilopascals to 2.5 kilopascals, 80% to 55% relative humidity at 36 degrees Celsius, at T equals 0 minutes, we predict damped oscillations in GS, shown in Figure 3.3a, and guard cell ABA concentration, denoted ABA subscript G, shown in Figure 3.3b. After a second step increase in vapor pressure deficit from 2.5 kilopascals to 3.9 kilopascals, 55% to 30% relative humidity at 36 degrees Celsius, at T equals 100 minutes, we predict sustained oscillations in GS and ABA subscript G. The approximately 20 to 40-minute oscillation periods predicted by the model are governed primarily by the interplay of two rate processes in ABA autoregulation, the stress-driven ABA synthesis arm, V stress, and the negative feedback ABA degradation, V-. Supplementary information. Figure B8e shows that the oscillation period decreases with increasing stress-dependent synthesis, V stress, and supplementary information. Figure B9f shows that the period decreases with increasing negative feedback, V-. Such minute-scale ABA dynamics are biologically plausible. NCED3 and CYP707A1/3 are upregulated, and foliar ABA accumulates within minutes of turgor loss in angiosperms. To understand the basis of this transition to sustained oscillations, we have explored variants in our coupled HP-HA model. Importantly, we find that the fully coupled model shows this transition, as seen in figures B13a and B14a, whereas if we cut ABA-regulated osmoregulation, as for the OST1 mutant, both damped and persistent oscillations are eliminated, as shown in figures B13b and B14b, even if an equivalent perturbation is imposed on the HP dynamics via a step change in osmotic potential in the guard cells, as shown in figures B13c and B14c. Thus, the HP processes in our model cannot explain the observed tendency toward oscillations. On the other hand, when we directly perturb the concentration of ABA in the guard cell by imposing a perturbation in its stress-dependent synthesis rate, V stress, we retrieve both damped and persistent oscillations, as shown in figures B13d and B14d. Additionally, we find, as suggested by Cowan, that vapor pressure deficit acts as an amplifier of nonlinear response that leads to oscillations, the effect of which can be modulated by the strength of mechanobiological transduction, V stress as a function of delta Pg, shown in figures 3.1d through E and figure B8, and by the negative feedback parameter V-, which controls ABA catabolism via A8H, shown in figure 3.1e and figure B9d through F. We conclude that ABA autoregulation provides critical nonlinear feedback that, in our framework, explains the transition from damped to persistent oscillations with increasing evaporative demand. Section 3.4.4 Hysteresis in stomatal conductance emerges from ABA autoregulation. We next explored whether the nonlinearity encoded in ABA autoregulation can capture another important feature of stomatal regulation, namely the hysteretic response in steady-state stomatal aperture to cycles of water availability, that is, dry down and recovery. This effect has been proposed to serve as a stress memory that can protect plants from recurrent drought, as shown in Figure 3.1c. To generate the bistability, that is having two steady-state solutions, required for hysteresis, we used a distinct parameterization by strengthening ABA's positive feedback gain, V plus over V stress, roughly 1.7-fold, as shown in Figure 3.4a. This adjustment could represent a physiological shift from a naive, monostable to a primed, bistable state. While the system parametrized for transients operates in a monostable regime, experimental evidence confirms that prolonged drought can create the primed, bistable state via epigenetic remodeling, which increases the induction of key ABA synthesis genes upon subsequent stress exposure, thereby increasing the gain of the positive feedback loop in ABA autoregulation. With this adaptation of parameters, predictions of the model still show reasonable agreement with wild-type stomatal dynamics, but do not show persistent oscillations at elevated vapor pressure deficit. To simulate cycles of water availability, we varied xylem water potential, psi x xyl, keeping vapor pressure deficit constant at 1.1 kilopascals, and tracked the trajectories of steady-state GS and guard cell ABA concentration, denoted ABA subscript G, through dry down and recovery as shown in Figures 3.4b and 3.4c. For dry down, that is decreasing psi x xyl, GS remains high, and ABA subscript G remains low from the unstressed state, where psi x xyl equals 0 megapascals, to a moderate stressed state, where psi x xyl is approximately negative 0.7 megapascals, before transitioning to a low GS and high ABA subscript G state for continued lowering of psi x xyl. For recovery, the stomates remain in their low GS and high ABA subscript G state until near complete recovery has been achieved, where psi x xyl is approximately negative 0.1 megapascals, at which point the predicted state jumps back to the original high conductance state. Each steady state point requires approximately 25 to 50 minutes per step, a lower bound set by ABA autoregulation kinetics. Under realistic plant, soil, and environment conditions, the rate of psi x xyl change is instead governed by soil hydraulic conductivity, rooting depth, evaporative demand, and canopy conductance, placing the full dry-down recovery cycle on a timescale of days to weeks. We extract GS, guard cell ABA concentration, and the net rates of NCED, a proxy for the rate of ABA synthesis, and CYP707A, a proxy for the catabolic degradation of ABA for 4 states visited as follows: from a well-watered state, W1, on a dry down to a stressed state, S1, followed by recovery to a well-watered state, W2, and a second dry down to a stressed state, S2, as shown in Figure 3.4d through G. We note that while the water potentials for the 2 well-watered states were the same, psi x xyl approximately negative 0.2 megapascals, the predicted macroscopic state of the stomates and the underlying molecular states were different for W1 and W2, reflecting differences in the 2 history-dependent solutions to the model equations. We note an important distinction between kinetic and bistable hysteresis. The kinetic mechanism, in which slow catabolism of ABA would cause a temporal lag in stomatal reopening, is inconsistent with experimentally measured rates of ABA degradation, gamma sub A approximately 0.25 per second, which predict clearance of ABA on minute timescales. In our bistable model, hysteresis arises from a mathematical property of the governing equations. The nonlinear positive feedback of ABA on its own synthesis produces an S-shaped steady-state ABA concentration curve with 2 stable states for the same psi x xyl. During recovery from drought, the system remains in a state with low stomatal conductance and high ABA because positive feedback sustains synthesis above catabolism. Both synthesis and catabolism are elevated, but synthesis dominates, and stomata reopen only when psi x xyl recovers past the bifurcation point, collapsing the high ABA state entirely. This steady-state bistability, not kinetic lag, is the mechanism of hysteresis predicted here. The experimental data of Verlouvet and Fromm, in which stomatal aperture differences persisted after 24-hour equilibration at the rewatered state, orders of magnitude longer than ABA clearance timescales, are consistent with this steady-state interpretation. Importantly, These predictions of hysteretic behavior across scales, from ABA-related processes in guard cells to stomatal regulation, agree semi-quantitatively with the molecular and macroscopic measurements reported by Verlouvet and Fromm in Arabidopsis. The autoregulation of ABA in guard cells drives this hysteresis in our model. Although previous investigators have suggested a role for ABA in generating hysteresis, no predictive models of this process have been put forward, and more generally, we are unaware of models of stomatal response that capture this potentially critical form of stress memory and adaptation to long-term variations in drought stress. Finally, our model predicts that this hysteresis is not universal. Steady-state memory requires the Bistable regime, entered only when positive feedback is strong enough to sustain elevated ABA through prolonged stress. Species in which foliar ABA instead rises and then declines during drought would remain in the monostable regime, where no hysteresis occurs, a testable cross-species prediction, consistent with the divergent drought responses reported among conifers. Section 3.5, Discussion. Our coupled hydropassive hydroactive, or HPHA, model builds on 50 years of developments of hydraulic models and, for the first time, introduces a framework to integrate the richness of genetic, molecular, and membrane processes by which ABA could modulate stomatal conductance. With this model, we identify guard cell localized autoregulation of abscisic acid, ABA, as a central driver of HA stomatal responses, including the emergent phenomena of damped and sustained oscillations and of hysteresis during drought and recovery. This localization provides a unified physical basis for responses to changes in water availability and demand without invoking elusive humidity sensors, explaining both the rapid response to water demand, VPD, and the role of hysteresis as a functional stress memory that anticipates recurrent deficits in water availability, PSIXYL. While not relevant to the existing data in AT used here, we note that our formulation of HP processes allows for accounting for newly elucidated non-stomatal regulation of transpiration by explicitly modeling the evaporative processes within the leaf. This framework provides new avenues to pursue important outstanding questions about stomatal responses to water stress and demand by allowing for the encoding of genotype-specific molecular and tissue-scale hypotheses. For example, while we show that mechanobiological coupling of guard cell turgor to guard cell localized upregulation of ABA, V-stress of delta PG, can explain observed stomatal dynamics in AT in a manner consistent with existing measurements of foliar ABA in other species and regulation of ABA synthesis, the framework also generates specific, falsifiable predictions. For example, that oscillations require intact ABA autoregulation and emerge only above a quantifiable stress threshold, or that guard cell-localized mechanotransduction and ABA synthesis suffice to explain the observed dynamics, predictions that new experimental data can refine or reject. Particularly valuable data would include simultaneous, real-time measurements of cytosolic ABA concentration via FR-ET-based reporters and guard cell turgor via pressure probe and spatially resolved flux measurements of ABA to deconstruct the V-stress term using guard cell-specific promoters driving reporters for key genes, for example NCED and CYP707A, to quantify their relative in vivo contributions during a stress response. As with the transient responses driven by ABA, we have not excluded that mesophyll-hosted regulation of ABA could undergo the same adaptation to drought as we have hypothesized in the guard cells, and thus contribute to hysteresis. Distinguishing between the contributions of these sites of ABA regulation remains an important outstanding challenge for the field. More broadly, like other mechanistic models of stomatal regulation, the model presented here remains underconstrained by the data currently available. Several parameter sets reproduce the present observations, and discriminating among them will require the guard cell resolved measurements outlined above. The model presented here focuses on water stress responses mediated by ABA and does not incorporate carbon assimilation or its activation of SLAC1 through the CO2 pathway. However, the CO2 pathway acts as negative feedback on stomatal closure, as ABA-driven closure reduces GS. CI falls, where CI equals CA minus 1.6 times A divided by GS. Guard cell H, CO3 minus declines, and the CO2/HCO3 minus driven contribution to SLAC1 gating diminishes. We assessed the magnitude of this effect by extending the model to include CO2-dependent SLAC1 gating, with the CO2 and ABA inputs combining multiplicatively, with the full derivation in SSI section B 6.4. The inclusion of this pathway produces only modest shifts in the predicted transients, approximately 37% versus approximately 43% closure, with versus without CO2 feedback, and does not alter the qualitative WWR and right-way response RWR features, or the bifurcation structure governing oscillations and hysteresis. We do not, however, rule out broader influences of photosynthesis when A varies dynamically. Integration of our framework with complementary models such as the OnGuard family could be of significant value for studying water use efficiency from both water and carbon perspectives. Such integration would extend naturally to the comprehensive treatments of ion transport and metabolic networks for light and CO2-driven responses in the OnGuard models, and to hypotheses of stomate-stomate coupling, such as proposed by Maud and Peake. Such integration would yield a unified, multi-signal systems model capable of quantifying, for example, how ABA-driven stomatal closure competes with light-driven opening, how CO2 modulates the ABA pathway, and how fully detailed single-cell processes can couple to define whole-leaf spatial and temporal patterning of stomatal dynamics to address collective dynamics like stomatal patchiness. Further integration with models of photosynthesis would form an unprecedented basis to pursue criteria for optimal stomatal function, providing a mechanistic foundation for the long-standing ideas about evolutionary optima and for the bioengineering of programmable plants that allow for resource use-efficient crop production. Finally, scaling this molecular-to-tissue framework to canopy processes would allow for mechanistic prediction of ecosystem-level plant-atmosphere coupling that is strongly controlled by stomatal conductance. Acknowledgements. We thank F. E. Rockwell, V. Bachiva, S. Sen, I. Gabay, E. Wu, J. Belding, and P. Jain for insightful discussions. This work was supported by the Center for Research on Programmable Plant Systems, CROPS, a science and technology center of the National Science Foundation under grant number DBI-2019674, competing interests. None. Code availability. All Python code used to generate the results in this study is available on GitHub at https://github.com/desai-sahil/stomatal-conductance-model, and the interactive dashboards are available at https://desai-sahil.github.io/stomatal-conductance-model/. Refer to Appendix B, Section B7, for details on the steps to run the code to reproduce the results in the main text. A note for the listener. This thesis refers to appendix material, which is not included in the audiobook. You can find it in the full written text. Chapter 4. The Curious Case of Cotton Physiology. Section 4.1.1. The Outside Xylem Zone, AquaSheet, Phenotype and its Species Dependence. In Chapter 2, we demonstrated that the outside xylem zone hydraulic conductance, GOXZ, in maize, maize L, declines several-fold before the Turger loss point during progressive drought stress, generating large apoplastic water potential gradients between the xylem, C-xyl, and the substomatal cavity, SSC APO, that are undetectable by conventional gas exchange or pressure chamber methods. The ratio of stomatal conductance to OXZ conductance, GS divided by GOXZ, emerged as the key dimensionless quantity governing this phenotype. When the ratio is large, as observed in maize under moderate drought, intercellular airspace undersaturation becomes significant. Standard gas exchange instruments underestimate true GS and measurable intrinsic water use efficiency gains accrue from non-stomatal OXZ resistance. When the ratio is small, stomata dominate and the OXZ makes a negligible contribution to transpiration regulation. The multi-species dataset in Jain and colleagues revealed that this phenotype is not universal. In maize, a C4 species with a bundle sheath-centered leaf architecture, the non-stomatal OXZ effect is pronounced. In tomato, Solanum lycopersicum, a C3 species, GOXZ declined with Psi XIL, but remained large relative to GS, keeping GS divided by GOXZ near 0.01 to 0.02 across the drought trajectory and producing negligible IAS under saturation. Preliminary aquadust measurements in cotton, Gossypium hirsutum, another C3 species, wildtype, included in the supplemental data of Jain and colleagues, figure S9, similarly yielded GS, divided by GOXZ, approximately 0.015, placing cotton alongside tomato in the stomata-dominated regime and in sharp contrast to maize. This species contrast raises two questions central to the present work. First, whether the non-stomatal OXZ hydraulic phenotype observed in a C4 grass extends to a C3 crop species such as cotton, and second, under what genetic and environmental conditions, if any, this phenotype becomes apparent in cotton. Section 4.1.2, a 50-year debate about cotton. Cotton has occupied a historically distinctive and unresolved position in the internal airspace undersaturation debate. The question was first posed and first contested with cotton as the experimental subject. Jarvis and Sladier used an inert tracer gas in cotton leaves and inferred internal airspace relative humidity as low as 0.70 to 0.75 under high evaporative demand, attributing this to limited water transport through mesophyll cell walls. Their conclusion was explicit. The water vapor pressure at the evaporation sites is not equal to the saturation vapor pressure under all transpiring conditions, directly challenging the foundational assumption underpinning standard gas exchange analysis. Farker and Raschke contested this interpretation, arguing that the apparent mesophyll cell wall resistance reported by Jarvis and Sladier was largely an artifact of their experimental conditions, and their own measurements in cotton provided no evidence for a significant internal resistance to vapor transfer under physiologically relevant transpiring conditions. This Jarvis and Sladier versus Farker and Raschke disagreement, conducted entirely in cotton, framed the intercellular airspace undersaturation debate for nearly 5 decades and established cotton as the test species for this question. The debate was brought back into focus by Wang and colleagues, who returned to cotton using noble gas tracing and double-sided gas exchange, and measured internal airspace relative humidity as low as approximately 80% at high vapor pressure deficit. They proposed a vapor pressure deficit-driven, aquaporin-mediated mechanism. As evaporative demand increases, plasma membrane aquaporins in the mesophyll progressively restrict transmembrane water flux to maintain cytosolic water potential. This limitation on liquid supply to the evaporating surfaces causes the saturation edge to retreat into the internal airspace. The mechanism they propose is distinct from the Xyl-driven, vapor pressure deficit-independent OXZ conductance decline demonstrated in maize in Chapter 2, and Jain and colleagues yet both likely share the same cellular substrate, plasma membrane intrinsic proteins, or PIPs, and differ primarily in their environmental trigger. Whether both mechanisms operate in cotton under combined drought and high vapor pressure deficit, and whether they can be resolved experimentally, remains an open question. Section 4.1.3, GHSLAC1, Cotton Physiology, and the case for this study. The experimental strategy developed in Chapter 2 provides the tools to probe whether cotton possesses a non-stomatal OXZ hydraulic phenotype. In maize, the SILAC 1,2 null mutant lacks functional ZMSLAC1, the primary S-type anion channel, mediating ABA-induced guard cell anion efflux and hydroactive stomatal closure. Because the hydroactive pathway is genetically suppressed in SLAC1, This mutant maintains higher GS than wild type, pushing the ratio GS divided by GOX to larger values and making the non-stomatal phenotype more pronounced in SLAC1 under moderate drought. The functional orthologue in cotton, GHSLAC1, was characterized by Wren and colleagues, who showed that silencing GHSLAC1 in Gossypium hirsutum impairs ABA-induced stomatal closure and results in elevated GS and increased water loss under drought. A loss-of-function cotton SLAC1 mutant is therefore the direct genetic counterpart to maize SLAC1. By maintaining high GS under drought in a C3 background, it provides the experimental condition needed to ask whether cotton's GS divided by GOXZ can be driven into the non-stomatal regime. Gossypium hirsutum, cotton, accounts for more than 90% of global cotton production, and cotton as a whole averages about 25% of total world fiber use. It is grown predominantly in semi-arid, irrigated environments, where daytime VPD routinely exceeds 3 to 4 kilopascals and where drought at any stage of reproductive development reduces fiber yield and quality. Improving water use efficiency without sacrificing fiber output is a central breeding objective, yet existing approaches focus on ABA-mediated stomatal closure and root architecture. The non-stomatal OXZ hydraulic phenotype has not been characterized in cotton as a WUE mechanism, nor has it been linked to any genetic locus. This work is conducted in collaboration with Dr. Andrew Nelson at the Boyce Thompson Institute, whose group has demonstrated that drought response and fiber yield maintenance in upland cotton can be linked to specific genetic variation. Building on that foundation, this chapter focuses on the physiological characterization of the outside xylem hydraulic phenotype, using wild-type and GHSLAC1, mutant cotton, and the ratio GS divided by GOXZ to determine when and whether a non-stomatal phenotype emerges. Leaf tissue has been collected in parallel for transcriptomic analysis, but those genomic data and their integration with the physiological traits lie outside the scope of this chapter. Demonstrating a non-stomatal phenotype in cotton and associating it with a defined genetic background would establish a mechanistically grounded, tractable target for future genetic studies aimed at achieving IWUE gains through OXZ resistance, rather than restriction of carbon assimilation. Section 4.1.4, Chapter Scope and Organization. This chapter uses aquadust-based measurements of leaf water potential and steady-state gas exchange in wild-type and GHSLAC1 mutant cotton to identify when stomatal and outside xylem hydraulic limitations to transpiration emerge during water stress, and how those limitations depend on xylem water potential and evaporative demand. The first aim is to characterize, in a directly measured and genotype-resolved way, the stomatal and non-stomatal responses of cotton to progressive water stress. Using aquadust together with steady-state LI-Core 6800 gas exchange, we quantify GS, PSISS-CAPO, GOXZ, and the ratio GS divided by GOXZ in wild-type and GHSLAC1 mutant plants across a soil drying trajectory, parameterized by PSI-XYL, and a range of vapor pressure deficits, or VPD. This extends the preliminary wild-type cotton aquadust measurements reported by Jane and others to a full drought trajectory and a SLAC1 genetic background, allowing us to ask when, if at all, cotton shows a significant OXZ resistance contribution to transpiration control. The second aim is to determine under what combinations of drought stress and evaporative demand the OXZ resistance phenotype becomes important. By independently varying Psi-XIL and VPD, we test whether cotton's OXZ limitation is primarily Psi-XIL-driven, as in the maize results of Chapter 2, VPD-driven, as proposed by Wang and others, or reflects the simultaneous operation of both mechanisms. The remainder of the chapter is organized as follows. Section 4.2, Materials and Methods, describes the plant material, growth conditions, drought and VPD treatments, aquadust usage protocol, gas exchange measurements, and data analysis procedures. Section 4.3, Results, presents the stomatal and OXI responses of wild-type and SLAC1 cotton, organized around the GS/GOXZ framework and the separation of CISI-XYL and VPD effects. Section 4.4, Discussion, interprets these results in the context of the IAS undersaturation debate in cotton, compares the cotton phenotype to maize and tomato, and considers implications for future genetic dissection and breeding for water use efficiency. Section 4.2, Materials and Methods. Section 4.2.1, Plant Material and Growth Conditions. 8. Experiments were conducted on cotton, Gossypium hirsutum, wild-type plants, and a GH/SLAC, one loss of function line, SLAC1, provided by the laboratory of Dr. Andrew Nelson at the Boyce Thompson Institute, BTI, Ithaca, NY, USA. All plants were grown from seed in pots under standard BTI greenhouse conditions, with controlled temperature, relative humidity, and supplemental lighting to maintain vegetative growth. Measurements were made on the uppermost fully expanded main stem leaf in flowering plants. Section 4.2.2, Drought and VPD Treatments. Progressive soil drying was imposed by withholding irrigation over several days prior to measurement, following the approach used in maize and tomato. On each measurement day, plants were sampled across a range of xylem water potentials, CSIG-XYL, spanning well-watered conditions to values approaching the leaf turgor loss point, and a range of VPD, as determined in preliminary measurements. Within this range, we targeted CSIG-XYL bins for low water stress, CSIG-XYL greater than or equal to negative 0.8 megapascals, and high water stress, CSIG-XYL less than negative 0.8 megapascals, to resolve the dependence of outside xylem conductance, GOXC, and the ratio GS divided by GOXC on water stress. Gas exchange measurements were performed between 900 and 1400 to limit diurnal variation. For each CSIG-XYL bin and genotype, plants were assigned to one of several leaf-to-air vapor pressure deficit, or VPD, conditions imposed in the gas exchange chamber, 2.5, 3.5, or 4.5 kilopascals, so that CSIG-XYL, set by soil water status, and VPD, set by chamber conditions, were varied independently. This design of experiment allowed us to separate the effects of xylem water stress and evaporative demand on the outside xylem phenotype. Section 4.2.3, Xylem Water Potential. Xylem water potential, CSIG-XYL, was determined from aquadust measurements in both sides taped leaf regions, calibrated against Scolander pressure chamber measurements. These pressure chamber values were used as a one-point calibration to map aquadust fret efficiency in taped regions to CSIG-XYL for all plants, exactly as implemented for maize in Chapter 2. Section 4.2.4, Aquadust Usage and Optical Measurements. Aquadust nanoparticles were synthesized and calibrated as described in Chapter 2. Aquadust, a polyacrylamide-based FRET hydrogel nanoreporter, was infiltrated into the target leaf with a blunt syringe to produce two approximately 1 to 2 centimeters squared infiltrated regions per leaf. One region was covered on both sides with optically transparent, gas-impermeable tape, the both sides taped, or BT, condition, to suppress local transpiration and report CSIG-XYL. The second region was left uncovered, the no tape, or NT, condition, so that the aquadust signal reported the apoplastic water potential at the evaporating surface in the outside axylem zone, referred to as Psi-SSC APO. This BT/NT geometry and interpretation follow Jane et al. Exactly. Local FRET emission spectra were collected with a fiber optic probe positioned at the leaf surface over each infiltrated region. Donor and acceptor emission intensities were used to calculate FRET efficiency, which was converted to water potential using the calibration curve established for cotton. For each BT and NT region, 3 to 5 spectra were averaged to obtain PSI-XYL in the BT region and CSI-SSC APO in the NT region, with gas exchange measurements. Measurements were made from the abaxial surface of the cotton leaf. Section 4.2.5, Gas Exchange Measurements. 8. Steady-state gas exchange was measured with a LI-Core 6800 portable photosynthesis system from LI-Core Biosciences, Lincoln, Nebraska, USA. The chamber enclosed the central portion of the same leaf bearing the aquadust regions. Unless otherwise noted, chamber conditions were photosynthetic photon flux density 600 mu mol-meters to the power of -2 seconds to the power of -1. Leaf temperature 36 degrees Celsius, CO2 concentration 420 mu-mol per mole, and leaf-to-air saturation deficit, referred to as SD-air, set between approximately 2.5 and 4.5 kilopascals. For each VPD set point and leaf, net CO2 assimilation, A, transpiration, E, and stomatal conductance, GS, were recorded once steady state was reached, defined as less than 1% coefficient of variation in E over a 2 to 3 minute interval. Across plants, CI axial and VPD were sampled independently across the desired ranges. Section 4.2.6, Calculation of outside xylem conductance. Outside xylem conductance, or GOXZ, was calculated by combining aquadust water potentials with gas exchange fluxes, following the equations and workflow in Jane et al. And Chapter 2. For each leaf, PSI-XYL from the BT region and CISSC-APO from the NT region were converted to corresponding water vapor mole fractions at the xylem, WXYL, and at the evaporating surface, WSSC-APO. The liquid phase conductance from the xylem to the evaporating surface was then computed as equation 4.1, where E is the total transpiration flux, corrected for boundary layer effects. For each observation, we computed the dimensionless ratio of GS to GOXZs as a function of PSI-XYL and used this ratio as the primary phenotype, describing the relative importance of stomatal versus outside xylem resistance to transpiration. Section 4.2.7, Statistical Analysis. Data processing, technical and biological replicate identification, and statistical analyses followed the workflow used in Jane et al. And Chapter 2. Xylem water potentials were grouped into two discrete bins, psi-axial greater than or equal to -0.8 megapascals and CI-axial less than -0.8 megapascals, to summarize the dependence of GS, PSI-SSC-APO, GOXZ, and GS divided by GOXZ on PSI-axial for each genotype. Within each bin, medians and interquartile ranges are reported. Differences between genotypes at a given PSI-XYL-VPD bin were assessed using Welch's t-tests with Benjamini-Hochberg correction to control the false discovery rate across multiple comparisons. Section 4.3.1, Light and CO2 response for SLAC1 cotton. Stomatal function in the GH-SLAC1. Loss of function, SLAC1 background was first assessed under standard light and CO2 perturbations. In the CO2 response protocol, cuvette CO2 concentration was stepped from 400 to 2000 mu-mol-mol to the -1 and back at fixed PAR, VPD, and cuvette temperature. Stomatal conductance in SLAC1 leaves remained high and nearly constant, approximately 220 to 250 millimoles per square meter per second, throughout the CO2 step, while net CO2 assimilation increased from approximately 19 to approximately 29 mu-mol per square meter per second at elevated CO2 and returned to the initial rate upon reestablishing 400 mu-mol-mol to the -1. This behavior is consistent with a loss of CO2-induced stomatal closure combined with intact photosynthetic biochemistry, as reported for SLAC1 mutants in Arabidopsis and maize. In the light response protocol, photosynthetically active radiation was stepped from 650 micromole meters to the power of -2 seconds to the power of -1 down to darkness at constant CO2, vapor pressure deficit, and cuvette temperature, as shown in Figure 4.1b. Stomatal conductance again showed only modest variation around a high baseline, while assimilation dropped from approximately 18 to 20 micromole meters to the power of -2 seconds to the power of -1 at highlight, down to 0 in the dark, as shown in Figure 4.1b, middle and lower panels. The combination of weak light and CO2 responsive stomatal conductance, alongside intact photosynthetic responses, is consistent with the role of GHSLAC1 as an essential mediator of ABA, CO2, and dark-induced stomatal closure in cotton guard cells. This characterization establishes that the cotton SLAC, one line used here, behaves as expected for a SLAC1 null background. Stomata remain relatively open over a wide range of light and CO2 conditions, while photosynthetic capacity is maintained. Section 4.3.2 Calibration of aquadust using Scolander pressure chamber. Aquadust nanoparticles were successfully infiltrated into leaves of both wild-type and SLAC1 cotton, yielding clean fluorescence emission spectra with clearly resolved donor, approximately 520 nanometers, and acceptor, approximately 560 nanometers, peaks in both genotypes. The signal-to-noise ratio of the spectra was comparable to those reported for maize and tomato, confirming that aquadust functions as a reliable apoplastic water potential reporter in cotton leaf tissue and that the infiltration protocol required no species-specific modification. Relative FRET efficiency increased with decreasing water potential for both genotypes, spanning from approximately 0.2 near negative 0.2 megapascals to approximately 0.5 near negative 15 megapascals. The calibration curves for wild-type and SLAC1 were similar in shape, but showed a slight genotype-specific offset. Each genotype's data was therefore evaluated using its own calibration curve to avoid cross-genotype error. To convert relative FRET efficiency to absolute water potential, aquadust readings from both sides taped, or BT, leaf regions were calibrated against Scolander pressure chamber measurements using a one-point calibration as implemented for maize in Chapter 2. The Scolander pressure chamber measurements and their paired FRET readings were used to construct the calibration curve for wild-type and SLAC1, as shown in Figure 4.2c. Together, these results establish that the aquadust Scolander pressure chamber framework, developed for maize, translates without modification to cotton, providing the calibrated water potential measurements needed for subsequent characterization of stomatal and non-stomatal responses. Section 4.3.3 Injection response in cotton. To verify that the mechanical act of injection does not itself perturb leaf gas exchange, we first compared A, E, and GS between water-injected and non-injected regions of the same cotton leaves, as shown in Figure 4.3d through F. None of the three variables differed significantly between conditions. A had a p-value of 0.09. E had a p-value of 0.18, and GS had a p-value of 0.18. This confirms that gasket-assisted pressure injection alone does not disturb stomatal aperture or photosynthetic capacity in cotton. When aquadust was injected under identical conditions, a selective effect on stomatal function was observed. Net CO2 assimilation was not significantly affected by aquadust injection, with a p-value of 0.42, indicating that the nanoparticles did not affect mesophyll photosynthetic capacity, whereas both transpiration and stomatal conductance were significantly reduced in aquadust-injected regions relative to non-injected regions on the same leaf. Transpiration with a p-value of 0.02 and stomatal conductance with a p-value of 0.03, demonstrating an aquadust-specific effect on stomatal aperture. Because all water potential measurements and all inferred quantities such as GS and GOXZs in subsequent sections are obtained from aquadust, it is important to note that the optical response of the reporter itself remains well-behaved. Relative FRET efficiencies derived from the fluorescent spectra can be consistently mapped onto independent Scolander pressure chamber measurements using a single parameter calibration, and the underlying raw spectra show reproducible donor-acceptor behavior across biological replicates. This internal consistency justifies continued use of aquadust in cotton, with the quantified impact of infiltration on GS kept in mind when interpreting GS divided by GOXZ across treatments. Section 4.3.4 Characterizing the stomatal and non-stomatal physiology in wild-type and SLAC1 cotton. In wild-type cotton, stomatal conductance, referred to as GS, the outside exilum water potential, referred to as PSISSC, APO, and outside exilum conductance, referred to as GOXC, all responded to combined gradients of xylem water potential, referred to as SIXYL, and vapor pressure deficit, referred to as VPD. At low water stress, where SIXYL was greater than or equal to negative 0.8 megapascals, GS remained high across the VPD range and showed only modest declines with increasing evaporative demand. As SIXYL declined below negative 0.8 megapascals, GS decreased, while CSISSC APO became more negative, indicating that wild-type stomata closed as both xylem tension and VPD increased. However, the closure and undersaturation are not significantly different across treatments. Over the same range, GOXZ declined but remained large enough relative to GS that GS divided by GOXZ generally stayed below approximately 0.05, keeping wild-type cotton in a stomata-dominated regime even at the driest SIXYL and VPD combinations measured. The SLAC1 mutant displayed a qualitatively different pattern for SIXYL and VPD compared to wild-type. Within each PSI-XYL bin, GS in SLAC1 was similar to wild-type at a given VPD, yet GS in SLAC1 declined as VPD increased, with a significant reduction at 4.5 kilopascals VPD. The corresponding PSISSC APO values for the higher PSI-XYL bin remained closer to saturation than those for the lower SSI-XYL class at all VPD levels. Outside xylem conductance, GOXZ in SLAC1 stayed relatively high at PSI-XYL greater than or equal to negative 0.8 megapascals and declined at PSI-XYL less than negative 0.8 megapascals across VPDs, but these differences were not statistically significant. Consequently, GS divided by GOXZ remained small throughout, not exceeding approximately 0.1, indicating that stomata continued to dominate the total resistance to water loss in SLAC1 cotton across all PSI-XYL and VPD combinations measured. Despite these genotype-specific patterns, the outside xylem hydraulic pathway behaved similarly in wild-type and SLAC1 across the SIXYL VPD space. In SLAC1, CSISSC APO followed SIXYL more strongly than VPD, and GOXZ declined only modestly as SIXYL became more negative, as shown in figures 4.4b, C, and 4.5b, C. Even though SISSC APO was more negative in SLAC1 at lower SIXYL, the resulting undersaturation was not large enough to cause a substantial additional loss of GOXZ. As a consequence, the ratio GS divided by GOXZ remained small in both wild-type and SLAC1, never exceeding values on the order of 0.1, as shown in figures 4.4d and 4.5d, indicating that cotton remained in a stomata-dominated regime across all SIXYL VPD combinations sampled. Cotton has long occupied a central place in the debate over whether the intercellular airspace remains saturated during transpiration, from the low relative humidities inferred by Jarvis and Sladier to the near-saturation conditions reported by Farquhar and Rashka, and the VPD-driven undersaturation proposed by Wang and colleagues. In this chapter, we combined aquadust with steady-state gas exchange in wild-type and GHSLAC1 loss of function, CELAC1, cotton to revisit this question in a SIXYL minus VPD framework, informed by the multi-species aquadust work in maize and tomato by Jane and colleagues, covered in Chapter 2. We found that SSC APO frequently fell several megapascals below SIXYL in both genotypes, providing direct evidence that the evaporating surfaces in cotton can operate well below saturation. Across the SIXYL minus VPD space we sampled, PSI-SSC APO tracked CSI-XIL more strongly than VPD in SLAC1, moving between low and high CSI-XIL bins produced larger changes in CSI-SSC APO than increasing VPD at fixed PSI-XIL, and many within bin VPD effects were not statistically significant. These results support the Jarvis-Sladier conclusion that undersaturation can occur in cotton, but point to Sahil rather than VPD as the dominant driver under our conditions, and show that Sahil-driven undersaturation can arise in a species where VPD-driven mechanisms have been documented. Placing cotton in the broader aquadust framework, our results put cotton in contrast to maize. In maize, GOXA declines by roughly an order of magnitude or more before the Turger loss point. SSC-APO reaches extremely negative values, and GS divided by GOXZ can approach approximately 0.3 to 0.5, such that non-stomatal OXZ resistance exerts strong control over transpiration. Here, in both wild-type and SLAC1 cotton, GOXZ decreased only modestly as PSI-XYL fell, remained within one order of magnitude of its well-watered value, and did not differ significantly between genotypes. Across all PSI-XYL and VPD combinations, GS divided by GOXZ stayed on the order of 10 to the power of -2 to 10 to the power of -1, never approaching maize-like values. Cotton thus exhibits measurable Psi-SSC-APO undersaturation, but its OXZ vulnerability rarely becomes large enough relative to GS to generate a strongly non-stomatal regime, implying that for cotton under the semi-arid Psi-XYL and VPD regimes examined here, transpiration remains primarily stomatal limited, and the standard assumption of stomata-dominated resistance would introduce smaller errors in IWUE than in maize. These physiological inferences rest on aquadust, which both enables and perturbs the measurements. Water injection controls showed that gasket-assisted pressure injection per se leaves A, E, and GS unchanged, whereas aquadust injection selectively reduces E and GS while leaving A intact, consistent with a modest, nanoparticle-specific, partial stomatal closure. At the same time, our calibration data show that aquadust's optical response is internally consistent. Raw spectra are reproducible across biological replicates, and relative Farret efficiencies map to Scolander pressure chamber PSI via a single parameter calibration. This combination implies that our GS and GOXZ estimates come from leaves with slightly reduced GS, but that the mapping from Farret to PSI-XYL and PSI-SSC-APO is unaffected. If anything, the injection artifact likely biases our GS/GOXZ values downward and makes our conclusions about weak non-stomatal limitation conservative. Taken together, these results extend the aquadust framework from maize to a major C3 crop, demonstrate that Psi-XYL-driven undersaturation can occur in cotton, and highlight a C3-C4 contrast in leaf hydraulic architecture. Maize readily enters a strongly non-stomatal regime under drought, whereas cotton remains strongly stomata-dominated. Future work should combine more sophisticated injection methods with expanded CI-XY LVPD sampling and additional genotypes to test whether the non-stomatal resistance phenotype in cotton can be tuned as a mechanistic lever for marginal water use efficiency gains. Chapter 5. Soil Drying Dynamics, a 1D transient simulation using Richard's equation and confrontation with AquaSheet. Wu, I.F., Eric P. Jane, S. Desai, V. Bachiva, S. Sen, A. Huber, and A.D. Strouk, 2026, an optical reporting patch for spatio-temporal measurements of water activity in complex environments. Analytical Chemistry, Volume 98, Issue 16, pages 11,865 through 11,877. DOI 10.1021/acs.analchem.5C08117. Author contribution: S. Desai designed and implemented the one-dimensional transient soil drying simulations based on Richards's equation and performed the confrontation of model predictions with AquaSheet soil-water potential measurements. Section 5.1.1 Water and Soil, the upstream boundary of plant water stress. 8. The movement of water through the soil-plant atmosphere continuum, known as the SPAC, is governed by a thermodynamic gradient in water potential, PSI, measured in megapascals, spanning several orders of magnitude from the wet soil matrix to the dry atmosphere. In this continuum, the soil represents the source. It is the node of highest, that is least negative, water potential, and it is from here that all water available to the plant ultimately originates. The remainder of the gradient through roots, xylem, leaf mesophil, outside xylem zone, or OXZ, and ultimately the stomatal pore is driven and constrained by the thermodynamic state of water in the soil. As soil dries, an increasingly negative psi soil reduces the driving force available to the plant at every subsequent node in the continuum. As soil dries, two physical processes unfold simultaneously. First, psi soil decreases, becoming more negative, reducing the chemical potential driving gradient toward the root surface. Second, the unsaturated hydraulic conductivity of the soil, K of psi, declines as the soil dries, by several orders of magnitude over a relatively modest drop in volumetric water content, as soil pores drain and the connected liquid network through the soil matrix becomes increasingly discontinuous and constricted. This simultaneous decline in both driving force and conductance originates entirely upstream of the plant, in the pore network of the soil, before any xylem or stomatal response has occurred. A key question in plant water stress biology is where, along the SPAC, hydraulic limitation first manifests under drying conditions. There has been a long-standing tendency to frame drought stress primarily in terms of xylem or leaf-level responses, such as xylem embolism, loss of turgor, and stomatal closure. In this view, the soil is often treated as a boundary condition imposed in experiments. Recent work has begun to revise this picture. Burt et al., using neutron radiography combined with hydraulic modeling across contrasting soil textures, showed that the onset of stomatal downregulation is initiated not by internal hydraulic failure in the xylem or mesophil, but at the root-soil interface. As the soil dries locally around the root, the drop in K of psi prevents the bulk soil from resupplying water fast enough to match transpirational demand. And this rhizosphere hydraulic limitation is the first signal of water stress transmitted to the plant. This finding positions CI soil and its spatiotemporal evolution during drying as a primary driver of the stress cascade that ultimately governs stomatal behavior and photosynthetic assimilation. Understanding and predicting how CI soil evolves in space and time during a drying event is therefore not a peripheral concern for plant physiologists, but a central one. It influences when stress begins and whether the plant experiences a gradual, manageable depletion or an abrupt hydraulic disconnection. These distinctions matter not only mechanistically, but practically. Experimental systems that impose drought without characterizing psi soil cannot distinguish between treatments that differ in stress intensity. Section 5.1.2, Quantifying Soil Water Stress, a Persistent Gap. Despite the established importance of soil water potential as the upstream driver of plant stress, psi soil remains the least rigorously characterized variable in the drought biology literature. This gap has been identified at multiple levels, from experimental design to community-wide reproducibility, and it represents one of the most important outstanding methodological problems in the field. Versluis, Dwenger, and colleagues, in a multi-laboratory synthesis of 15 unresolved questions in plantibiotic stress biology, explicitly named the difficulty of connecting laboratory-imposed drought phenotypes to physiologically and ecologically meaningful stress levels as one of the central open problems. A recurring source of this difficulty is the practice of imposing drought by withholding water and tracking soil water content, theta, in units of meters cubed per meters cubed, rather than soil water potential. Theta is a volumetric quantity that conflates the thermodynamic state of water with soil texture-specific retention properties, making it non-transferable across soil types and, crucially, non-interpretable in terms of the thermodynamic energy available to the plant. Two soils at identical theta can differ by orders of magnitude in psi soil depending on their pore size distribution. Consistent with this, Van Buren and colleagues, in a reanalysis of hundreds of drought transcriptomic datasets, found that a large fraction of studies report no direct measure of plant water status, and when soil conditions are described, they are most often expressed only in terms of theta rather than psi soil, leading to pervasive interstudy variability in actual stress intensity that cannot be attributed to biology alone. Hence, to understand natural variation in drought responses, rigorous characterization of the soil stress is a key missing element and calls for broader adoption of psi soil measurements. The challenge of measuring CI soil is compounded in the field by pronounced spatial and temporal heterogeneity. Psi soil varies with depth and over timescales from hours following precipitation or irrigation to weeks during seasonal drying. Point measurements from tensiometers or thermocouple psychrometers provide snapshots at specific depths and locations that may be unrepresentative of the integrated signal experienced by the root system. Soil moisture heterogeneity has been documented from pore to landscape scales, so even well-instrumented field experiments typically yield psi soil data that are spatially incomplete. There is therefore a compelling need for tools, both experimental and computational, that can resolve the spatio-temporal evolution of psi soil with sufficient resolution to couple it meaningfully to plant physiological responses. AquaSheet, an optical reporting patch developed by Wu and others in our laboratory, provides the experimental platform for spatially resolved psi soil measurement, and this chapter uses a physics-based one-dimensional transient soil drying model based on Richard's equation to confront those measurements with quantitative predictions. Section 5.1.3, Chapter Scope and Organization. This chapter links experimentally measured soil water potential to a physics-based simulation of soil drying in order to quantify the upstream hydraulic boundary condition experienced by plants. AquaSheet, an optical reporting patch developed by Wu and others in the Stroke Laboratory at Cornell University, provides spatially resolved measurements of psi soil at the surface of a drying soil column, yielding time series of the thermodynamic variable that governs water flow in the soil-plant atmosphere continuum. The core aim of the chapter is not to develop new soil physics theory, nor to recharacterize AquaSheet, but to confront independent experimental and computational views of psi soil using a one-dimensional transient soil drying model based on Richards's equation, with isothermal Philip de Vries vapor conductivity, parameterized for the soils used in the AquaSheet experiments using the Van Genuchten formulation, and to assess how well this standard continuum framework captures the observed drying dynamics under controlled conditions. Two AquaSheet datasets are analyzed. In one, the model reproduces the spatio-temporal evolution of psi soil. In the other, it fails to do so. The divergence between model and measurement is used to identify which physical mechanisms and parameter regimes are most critical for accurately representing soil drying. The remainder of the chapter is organized as follows. Section 5.2, Materials and Methods, describes the soil properties, column geometry, AquaSheet deployment, the governing equations, and the numerical implementation of the one-dimensional simulation. Section 5.3, Results, presents the model predictions alongside the AquaSheet psi soil time series for both datasets. Section 5.4, Discussion, analyses the sources of agreement and discrepancy between model and experiment, with particular emphasis on the roles of liquid and vapor conductivity and on the limitations of the one-dimensional isothermal framework. Section 5.2, Methods. Section 5.2.1, AquaSheet, optical sensor for spatially resolved psi soil measurement. AquaSheet is a composite PDMS-based optical reporting patch that measures local water activity, AW, dimensionless, via a ratiometric fluorescence signal from embedded OG488 and rhodamine sensing elements. This design enables calibration-stable spatio-temporal tracking of AW, which is converted to water potential through the Kelvin equation, Equation 5.1. AquaSheet therefore reports water potential directly. The psi values measured in this work lie well beyond the operating range of standard soil sensors, such as tensiometers and psychrometers. For the experimental dataset reported here, AquaSheet was mounted on a black PDMS optical insulation layer to minimize background from the soil. Owing to its equilibration timescale of order 10 squared seconds, it can be treated as a quasi-steady reporter of SI soil on the hours to days timescale of soil drying. Full details of fabrication, calibration, and imaging are given in Wu et al. Section 5.2.2, Soil Composition, Column Geometry, and Experimental Setup. The soil used in all drying experiments was a binary mixture of approximately 1.5 grams of sandy loam and 1.5 grams of play sand, brand name Sacrete, mixed dry, combined with approximately 5 milliliters of deionized water and packed into a custom-built optical cell. The packed column was equilibrated overnight at AW approximately 1.0 prior to experiments. AquaSheet was placed on the soil surface, sandwiched between a plexiglass lid and the black PDMS optical insulation layer, as described in Figure 5A of Wu et al., and the simulation domain was taken to coincide with the AquaSheet ROI, which is 10.3 millimeters. Drying was imposed inside a custom RH-controlled box with ambient AW monitored by a hygrometer, and 2 datasets were collected under different ambient conditions. Fluorescence images were acquired and processed using the ratiometric imaging protocol of Wu et al., yielding spatially resolved AW of X and T maps that were converted to psi of X and T via the Kelvin equation. Equation 5.1. Section 5.2.3, Governing Equations. Richards' equation, Schramm form. Richards's equation, written in the water potential, psi, form, describes the dynamics of water transport in the unsaturated soil column. Equation 5.2, where C of psi equals partial theta over partial psi in units of inverse megapascals is the hydraulic capacitance, and K of psi in units of meters squared per second per megapascal is the total effective hydraulic conductivity of the soil. The psi form is adopted here because psi is the primary variable reported by AquaSheet. Water retention curve, Van Genuchten parameterization. The constitutive relationship between psi and volumetric water content theta in units of meters cubed per meters cubed is provided by the Van Genuchten parameterization of the water retention curve, abbreviated WRC, equation 5.3. Where theta dimensionless is the dimensionless effective saturation, theta R and theta S are the residual and saturated water contents, and alpha, in units of MPa to the power of -1, n dimensionless, and m equals 1 minus 1 over n dimensionless, are empirical shape parameters describing the soil pore size distribution. For a reader familiar with plant biophysics, the WRC plays an analogous role to the pressure volume, or PV, curve in plant cell mechanics. It is the nonlinear constitutive relationship between a thermodynamic state variable, psi, and a volumetric state variable, theta, that must be known before the governing transport equations can be solved. The hydraulic capacitance is obtained analytically by differentiating the water retention curve. Equation 5.4, Liquid hydraulic conductivity, Van Genuchten expression. The liquid phase hydraulic conductivity, KL, as a function of psi, is derived from the water retention curve via the Van Genuchten pore connectivity model. Equation 5.5, where Ks, in meters per second, is the saturated hydraulic conductivity. KL spans approximately 6 orders of magnitude between saturation, where theta equals 1, and air-dry conditions, where theta approaches 0. This is the primary physical reason why soil drying is not a simple linear diffusion process, and why numerical solution is required. Vapor hydraulic conductivity, isothermal Philip de Vries framework. 8. As the continuous liquid network breaks down at low water contents, vapor phase diffusion through the air-filled pore space becomes an increasingly important transport pathway. The vapor contribution to hydraulic conductivity is described using the isothermal limit of the Philip and de Vries framework, equation 5.6, where d-atm is the molecular diffusivity of water vapor in free air, tau is the tortuosity factor, Theta A equals theta S minus theta L is the air-filled porosity. Nu equals PATM divided by the quantity. PATM minus PSAT is the mass flow factor. RHOV and ρL are the vapor and liquid water densities, and R gas is the specific gas constant for water vapor. The Philip de Vries framework was originally developed in the context of thermally driven vapor transport, with coupled thermal and isothermal terms. The isothermal limit adopted here is appropriate to the controlled laboratory conditions of these experiments, where temperature gradients across the soil column are negligible. The total effective hydraulic conductivity is equation 5.7, with KL dominating near saturation and KV becoming dominant as the soil dries into the low water content regime. Section 5.2.4, Boundary and Initial Conditions. At the left boundary, where x equals 0 and the soil surface is exposed to the atmosphere, a Robin flux condition applies. The surface evaporative flux is parameterized by a surface vapor transfer coefficient H in units of meters per second per megapascal, which characterizes resistance to vapor transport across the soil-atmosphere interface. Equation 5.8. At the right boundary, where x equals L, representing the sealed base of the column, we apply a zero-flux Newman condition. Equation 5.9. The initial condition, the initial water potential profile, psi of x at time equals 0, was derived directly from the first AquaSheet image at time equals 0, converted from raw water activity using the Kelvin equation, equation 5.1. The raw profile was smoothed using piecewise cubic interpolation, followed by a moving mean filter with a window of 100 spatial points to remove high-frequency measurement noise while preserving the large-scale gradient, and was passed as the initial condition to the PDE solver. Section 5.2.5, Soil Hydraulic Parameters. All Van Genuchten and transport parameters were taken from Wu et al. And are summarized in Table 5.1. A table is shown here. Section 5.2.6, Numerical Implementation. The psi-form-Richards equation with the constitutive functions above was solved numerically using MATLAB's PDE-PE solver, which implements an adaptive finite difference scheme for one-dimensional parabolic partial differential equations. The spatial grid was defined by the AquaSheet imaging coordinates, spanning the 10.3 mm region of interest. The solver was run with Cartesian geometry. Simulation outputs, CI of x and t, were evaluated at the same time points as the experimental data and compared directly to the AquaSheet-derived CI of x and t profiles. All code used for the simulation is available at the web address listed in the thesis. Section 5.3.1, Hydraulic Constitutive Functions for Soil. The Van Genuchten water retention curve, shown in Figure 5.2a for the soil characterized in Table 5.1, shows a steep drop in volumetric water content theta over the psi range, and a rapid approach to the residual water content theta r as psi becomes more negative. The corresponding hydraulic capacitance C of psi, shown in Figure 5.2b, peaks near the air entry potential. The liquid phase hydraulic conductivity KL of Sasai spans roughly 6 orders of magnitude between saturation and the near residual regime, while the vapor phase conductivity KV of Sasai, computed from the isothermal Philip de Vries framework, is negligible in the wet range, but becomes comparable to and then exceeds KL as psi, decreases below approximately -1 to -5 megapascals, as shown in Figure 5.2c. This crossover indicates that for the psi range probed in the AquaSheet experiments, down to approximately -30 megapascals at the surface, vapor transport should play a significant role in the drying dynamics. Section 5.3.2, Dataset 1, Apparent Agreement with Liquid-Only Conductivity. In Dataset 1, the soil column dried from an initial psi profile of around -3 to -4 megapascals to a final state where surface psi approached approximately -15 megapascals over 50 hours, while the base remained substantially wetter. When the Richards equation is solved using only the liquid phase conductivity K sub L of psi, The simulation reproduces the main qualitative features of the measured psi of X and T profiles: the position and curvature of the drying front, the gradual steepening of the psi gradient near the surface, and the approximate time at which the profile begins to plateau. In contrast, when the vapor contribution K nu of psi is added with its physically realistic magnitude, so that K equals KL plus K nu, as shown in figure 5.3b, the model dries too rapidly and produces psi of X and T profiles that are substantially more negative and more spatially uniform than observed. At intermediate and late times, the simulated psi towards the base overshoots the measured values by several megapascals, and the predicted gradient into the column is much weaker than the experimental gradient. Thus, for dataset 1, the inclusion of vapor conductivity in the standard Philip de Vries form degrades the match to the AquaSheet data relative to the liquid-only model. Section 5.3.3. Dataset 2. Breakdown in the extreme dry regime. Dataset 2 represents a more severe drying experiment. The ambient water activity was lower, and the drying period was extended to 120 hours. AquaSheet measurements show that psi at the surface decreased from roughly -3 megapascals at T equals 0 to approximately -30 megapascals at Tt equals 120 hours, with a persistent gradient to less negative psi values deeper in the column. Using only the liquid conductivity KL of psi, the model again yields profiles that qualitatively resemble the measured drying fronts. Psi at the surface becomes steadily more negative. The front migrates downward with time, and the simulated profiles fall within a few megapascals of the experimental curves. Once the vapor pathway K nu of psi is included, as shown in figure 5.4b, the simulation for dataset 2 initially tracks the measured psi of X, T profiles reasonably well at early times, but then diverges in the extreme dry regime. As CCI becomes strongly negative and transport is dominated by vapor conductivity, the model produces profiles that are too uniform and insufficiently curved near the surface, with a drying front that advances too rapidly and deeply into the column relative to the AquaSheet data. The mismatch is most pronounced at the latest time points, where AquaSheet records psi values between -20 and -30 megapascals. Across both experiments, a Richards model with liquid-only conductivity KL of Ci can be tuned to give visually convincing fits to the AquaSheet psi of X and T profiles. But this apparent success disappears once the vapor pathway K nu of Psi is added with a magnitude derived from the Philip de Vries framework. The shift from acceptable fits under a liquid-only assumption to discrepancies with KL plus K nu is most pronounced in dataset 2, where average psi reaches more negative values compared to dataset 1, and the system operates entirely outside the calibration range of conventional soil hydraulic measurements. Taken at face value, the liquid-only simulation suggests that a simple Richards model can describe the observed soil drying dynamics in both experiments, even as Ci at the surface approaches -30 megapascals. This agreement, however, is achieved by effectively suppressing vapor transport. With Kv set to 0, the effective diffusivity d of psi, equal to Kl of psi divided by C of psi, becomes extremely small in the dry regime, and modest adjustments to Ks and the surface transfer coefficient H are sufficient to match the apparent speed and curvature of the drying front. In other words, the acceptable fit arises because a one-pathway model with tunable parameters is flexible enough to reproduce the observed psi as a function of X and T, not because the assumed theta of psi, C of psi, and KL of psi necessarily represent the true soil hydraulic behavior. When vapor conductivity is enforced using the Philip de Vries formulation and measured soil parameters, the model no longer has this freedom. In the dry regime, K nu of psi dominates KL of psi, and D of psi is set primarily by vapor diffusion through the air-filled pore space. Under these conditions, the one-dimensional isothermal Richards-Philip de Vries model fails to reproduce the AquaSheet profiles, especially in dataset 2, where psi falls below -20 megapascals. This breakdown is better interpreted as a diagnostic of where standard assumptions cease to hold than as a failure of Richards's equation itself. First, the van Genuchten parameters used here were fitted over the standard range of psi, typically down to about -1.5 megapascals, as is common in soil physics and plant physiology. Extrapolating this water retention curve to -20 or -30 megapascals likely misrepresents both theta as a function of psi and its derivative c as a function of ci, which in turn affects the inferred kl as a function of psi and k nu as a function of psi. AquaSheet, by directly measuring CI in this previously unconstrained regime, exposes this deficiency. Second, the model assumes a homogeneous soil column and strictly one-dimensional transport, while the actual system may exhibit vertical variations in packing, local textural heterogeneity, or preferential flow paths that effectively reduce the macroscopic vapor diffusivity relative to the idealized Philip de Vries prediction. Such heterogeneities would slow the spread of the drying front, bringing the experimental profiles closer to the liquid-only model than to the fully coupled KL plus K nu prediction. Methodologically, the comparison between AquaSheet measurements and the one-dimensional Richards-Philip de Vries model highlights why direct psi measurements extending well below the conventional wilting point, around -1.5 megapascals, are essential. Without such measurements, soil hydraulic models can be tuned to fit moderate stress data, while failing in the more negative psi regime that might be increasingly relevant for drought experiments. AquaSheet provides this missing resolution of CI soil down to -30 megapascals at sub-centimeter spatial scale and multi-day timescale, possibly highlighting where standard van Genuchten Philip de Vries parameterizations cease to be reliable. For plant hydraulics, these findings imply that models using such parameterizations to describe the root zone soil may misestimate the hydraulic boundary conditions experienced by roots, and consequently the stomatal safety margins and embolism risk inferred from them. If vapor and liquid contributions are treated inconsistently, for example by omitting them or by basing them on extrapolated constitutive relations, models may misestimate near-surface drying and misplace the onset of hydraulic limitation. In this chapter, the divergence between liquid-only and liquid plus vapor simulations and AquaSheet measurements is therefore not interpreted as evidence for the correctness of AquaSheet as a tool. Rather, it highlights that these simulations, based on extrapolated constitutive relations, cannot by themselves confirm or refute the accuracy of AquaSheet-derived measurements. Instead, it is a call to remeasure and reparameterize soil hydraulic properties in the extremely dry regime that AquaSheet now makes experimentally accessible. Chapter 6. Conclusions and Future Work. Section 6.1. Overview and Rationale. Over the preceding chapters, this thesis combined experimental tools and mechanistic modeling to dissect how stomata integrate hydraulic and biochemical signals from soil to leaf to regulate water loss under water stress. In maize, a hydropassive model informed by aquadust measurements and a SLAC 1 stomatal mutant quantified the contribution of pure hydraulics to stomatal regulation and highlighted further the role of a large outside xylem zone resistance that can dominate non-stomatal limitations to transpiration. In Arabidopsis, a HPHA model of guard cell autonomous abscisic acid, or ABA, synthesis and signaling showed that ABA autoregulation mediated through the PYR, PP2C, OST1, SLAC1 core signaling module can generate both damped and persistent oscillations in stomatal conductance and the hysteretic stress memory observed during drought and recovery. In cotton, aquadust-based characterization of outside xylem zone conductance and GS/GOX across genotypes and vapor pressure deficit demonstrated that a C3 crop can exhibit measurable intercellular airspace under saturation while remaining predominantly stomata-limited. Finally, a Richards equation soil model, coupled to AquaSheet measurements, showed that standard soil hydraulic parameterizations can fail dramatically in the extremely dry regime relevant to severe drought. In what follows, I organize future work into 4 interrelated themes: species-specific integration of hydropassive and ABA-mediated hydroactive modules, coupling ABA-driven stomatal regulation with the OnGuard platform and A-GS modeling, extending the HP-HA framework to Ca2+, ROS, and long-distance signaling, and coupling soil drying with stomatal regulation to form a unified model of the soil-plant atmosphere continuum, or SPAC. Section 6.2, Species-Specific Unified Hydropassive Hydroactive Models. A key limitation of the current work is that the hydropassive model is parameterized and tested primarily in maize, whereas the ABA-HP-HA model is built around Arabidopsis data and genetic resources. This division was advantageous for leveraging species-specific tools, such as Aquadust and maize/1 for hydraulics, and Arabidopsis ABA mutants for understanding hydroactive stomatal regulation. But it leaves open how hydraulics and ABA interact within a single species to shape stomatal responses under water stress. The next step is to construct a unified HP-HA model in a single species, such as maize or cotton, in which both hydraulic and ABA modules are jointly constrained by genotype-resolved data. In maize, the existing hydropassive model treats mesophyll, subsidiary cells, and guard cells as lumped elements connected by hydraulic resistances and capacitances, with GS proportional to the turgor difference between guard and subsidiary cells. The model is currently calibrated against SLAC1 data, where HA regulation is genetically suppressed, and then used to infer the implicit HA contribution in wildtype. Future work could embed the ABA autoregulation and signaling module developed for Arabidopsis directly into this maize HP framework, creating a species-specific HP-HA model in which guard cell ABA synthesis, ABA positive feedback, and catabolism via CYP707A family enzymes control SLAC1 activation and guard cell osmotic potential. Such a model would explicitly connect the hydraulic variables measured by Aquadust, namely xylem and OXY water potentials, to ABA dynamics and would allow quantitative partitioning of maize drought responses into OXZ hydraulics versus ABA feedback under realistic field-like conditions. In cotton, the current Aquadust results show that despite measurable OXY undersaturation, GS divided by GOXY remains small, keeping cotton in a stomata-dominated regime across the sampled CIXYL-VPD space. A unified HP-HA model calibrated on wildtype and GHSLAC1 mutants could test whether species-specific differences in guard cell elasticity, OXZ vulnerability curves, or ABA feedback can explain why cotton rarely shifts into a strongly non-stomatal regime. Comparative modeling between maize and cotton would directly test the hypothesis that differences in OXZ, vulnerability, and HA gains underpin species contrasts in intrinsic water use efficiency. Section 6.3. Coupling ABA-driven stomatal regulation with OnGuard and AGS modeling. At present, the Arabidopsis ABA-HP-HA model in this thesis resolves ABA autoregulation, PYR-PP2-COST1, SLAC1 signaling, and coarse-grained guard cell electrophysiology, while treating calcium implicitly and focusing on potassium and chloride fluxes. The OnGuard platform developed by Blatt and colleagues provides a complementary guard cell membrane electrophysiology-focused framework. The original on-guard implementation and its companion systems dynamics analysis resolve plasma membrane and tonoplast ion channels and pumps, including calcium handling, detailed voltage dynamics, and metabolism of major osmotica under changing light and CO2. Subsequent extensions, often referred to as on-guard 2, bridged guard cell to leaf scales and introduced a relative water feed factor to represent leaf water status, allowing predictions of transpiration responses to humidity, temperature, and water availability without explicitly resolving the upstream hydraulic circuit. A logical future direction is to integrate these approaches by replacing on-guard's phenomenological hardwired ABA input, as in on-guard 3E, with a mechanistic ABA autoregulation module driven by guard cell turgor, and by coupling its electrophysiology to a hydraulically explicit hydropassive module. In such a hybrid ABA on-guard hydropassive model, step changes in VPD or PSI-XYL would first alter guard cell turgor via the hydropassive module, triggering stress-dependent ABA synthesis, V-stress of PG, ABA positive feedback, and catabolic negative feedback via A8H. ABA would then bind PYR receptors, inhibit PP2C, activate OST1, and thereby modulate SLAC1 and related channels within the on-guard core, leading to changes in membrane potential and guard cell osmotic potential. Because the hydropassive module explicitly resolves xylem and SSC apoplastic water potentials, this framework would link aquadust-measured SAIXYL and PSI-SSC APO directly to ABA dynamics, Ca2+ signaling, and ion transport, rather than treating water status as an external relative water feed factor. Existing on-guard models have focused primarily on light and CO2 responses and, notably, revealed a carbon memory whereby guard cell endomembrane Ca2+ ATPases led to carbon hysteresis. By contrast, the ABA hydropassive hydroactive model developed in this thesis predicts a complementary water stress memory arising from ABA autoregulation and its bistable response to xylem water potential during drought and recovery. Integrating these two lines of work would enable mechanistic exploration of how carbon and water stress memories interact. For example, how a history of fluctuating CO2 and light, encoded via Ca2+ dependent feedback, combines with a history of drought, encoded via ABA feedback, to shape subsequent stomatal responses, photosynthesis, and intrinsic water use efficiency. Embedding the integrated ABA on-guard HP module into leaf scale A GS models would provide a pathway from guard cell scale signaling to canopy scale carbon and water fluxes. At the leaf and canopy scales, most photosynthesis transpiration models still treat stomatal conductance using empirical formulations such as Ball, Berry, or Medlin models, which relate GS to photosynthetic assimilation, atmospheric CO2, and VPD via fitted parameters, and assume that stomata present the dominant resistance to water vapor flux, thereby neglecting losses of outside xylem conductance and intercellular airspace under saturation. The maize and cotton chapters in this thesis demonstrate that such assumptions can fail under moderate drought. In maize, GS divided by GOX8 can approach 0.3 to 0.5, causing substantial intercellular airspace under saturation and non-stomatal control of transpiration, whereas in cotton, GS divided by GOXZ remains small, but PSI-SSC APO can still fall several megapascals below PSI-XYL. Future work should therefore replace empirical GS models in GS frameworks with mechanistic predictions from ABA, on-guard, HP models that account for both hydraulic and biochemical state variables, and use these models to explore how different combinations of OXZ vulnerability, ABA feedback gains, and guard cell electrophysiological properties shape the coevolution of A, E, and A divided by E during soil drying and rewetting. By extending on-guard's original scope beyond light and CO2 to encompass ABA-mediated water stress responses, and by anchoring the model in explicit leaf and soil hydraulics, this future work would move toward truly ecophysiology-ready stomatal models. Such models could predict species-specific responses under a wider range of environmental conditions, including fluctuating VPD, CO2, light, and soil water status, and provide mechanistic insight into water use efficiency from both the water and assimilation perspectives. Section 6.4. Extending the HPHA framework to Ca2+, ROS, and long-distance signaling. The current ABAHP-HA model treats calcium 2+ implicitly, blackboxing its effects into coarse-grained rate laws for ion fluxes and membrane voltage while holding symplastic pH fixed. Calcium 2+ dynamics in guard cells, however, form an important secondary messenger system that amplifies and encodes information about ABA. These calcium 2+ signals act through a kinase branch parallel to the OST1 branch that converges on SLAC1. In the calcium 2+ dependent branch, CPKs directly phosphorylate the SLAC1 N-terminus and are themselves negatively regulated by PP2C phosphatases such as ABI1. The OSCA family of hyperosmolality-gated calcium 2+ permeable channels provides a mechanosensing entry point into this network. OSCA1 mediates osmotic stress-evoked cytosolic calcium 2+ increases needed for osmosensing, and OSCA1 mutants show attenuated regulation of transpiration under osmotic stress. Because our current V-stress, delta PG, formulation is agnostic about which molecular sensors convert turgor into a biochemical signal, incorporating an OSCA-like calcium 2+ module, where guard cell turgor changes computed by the HP module drive mechanosensitive calcium 2+ influx into the CPK branch, would allow calcium 2+ dependent effects on SLAC1 and related targets to be captured explicitly and analyzed via bifurcation tools, described in Appendix B, yielding testable hypotheses in OSCA1 and CPK mutants. A note for the listener. This thesis refers to appendix material, which is not included in the audiobook. You can find it in the full written text. A critical dimension of such an extension is the integration of guard cell Ca 2+ signaling with long-distance hydraulic signals, and with the experimental tools needed to constrain the resulting models. Recent work by Bacheva and colleagues introduces a unifying poroelastic framework that captures both local and systemic hydraulic responses to wounding and predicts psi-XYL, as a function of time, relaxation dynamics that depend on transpiration. Feeding these poroelastic psi-XYL of T trajectories into the HPHA Ca2+ model would create a mechanistic bridge between whole plant hydraulic states and guard cell responses, in which Ca2+ signals and ABA synthesis respond explicitly to plant-scale hydraulics, enabling predictions for soil drought, which is slow and abiotic, wounding, which is fast and biotic, or their combinations. Testing these mechanistic models requires tools that can simultaneously report Ca2+ dynamics and physiological output with sufficient spatial and temporal resolution. Genetically encoded Ca2+ reporter plants now make it possible to visualize stimulus-specific Ca2+ transients in vivo across a range of timescales and spatial scales. Combining these Ca2+ reporters with aquadust measurements and Li-COR-based gas exchange would create a phenotyping platform in which molecular Ca2+ signals are directly correlated with the hydraulic and physiological state variables that drive the model, providing the observational constraints needed for parameter inference and validation. Reactive oxygen species, or ROS, are another major axis of guard cell signaling that the current HP-HA model does not resolve explicitly. ABA activates the NADPH oxidases ATRB-OHD and ATRB-OHF in guard cells, generating apoplastic H2O2 bursts that in turn activate calcium-permeable channels, modulate SLAC1, and drive stomatal closure. More recently, work from the Woodson Laboratory at the University of Arizona has shown that chloroplast-derived ROS signals form a part of specialized signaling networks. These findings point to a richer ROS landscape in guard cells than is currently captured and motivate the inclusion of a minimal ROS module in the HP-HA framework, enabling exploration of combined stress scenarios such as heat plus drought, where ROS production is elevated independently of ABA, and prediction of how such stresses modify stomatal responses. Genetically encoded reporters of cellular ROS could be combined with the calcium imaging and AquaDust platforms described above to simultaneously monitor ROS, calcium, and hydraulic state. This multi-signal approach would provide rich observational constraints for the extended model and help dissect the specificity with which each molecular mechanism contributes to stomatal regulation, a central question in the rapidly expanding field of long-distance signaling in plants. Section 6.5, Coupling Soil Drying, Root Uptake, and Stomatal Regulation. The soil chapter shows that a standard one-dimensional Richards equation model with Van Genuchten retention and Philip de Vries vapor conductivity can be tuned to fit moderate soil drying data, but fails once soil dries beyond -20 to -30 megapascals at the surface. This breakdown reflects extrapolation of constitutive soil relations beyond their calibration range and omission of heterogeneity effects, and underscores that soil hydraulic behavior in the extremely dry regime remains poorly constrained. Independent work using neutron radiography and hydraulic modeling indicates that stomatal downregulation may be initiated by hydraulic limitations at the root-soil interface, rather than by xylem embolism or leaf-level events. A future direction is to couple the Richards-based soil model to a root water uptake module, and then to the HP-HA stomatal model, creating an integrated soil root xylem leaf framework constrained by AquaSheet and AquaDust measurements. AquaSheet-derived CI soil of Z and T profiles would feed into a root uptake model that accounts for root length density and rhizosphere conductivity, which in turn provides xylem boundary conditions to the leaf HP module. The HP-HA model would then predict GS, CSI-SSCAPO, ABA, CA2+, and ROS dynamics, closing the loop with transpiration and feeding back on soil drying. This integrated framework would allow studying the SPAC under different soils and atmospheric forcing. Section 6.6, Concluding Perspective. The modeling work in this thesis highlights the importance of rigorous parameter inference and identifiability analysis. The maize hydropassive model uses Markov chain Monte Carlo with Bayesian learning to quantify posterior distributions and parameter correlations, revealing which parameters are tightly constrained and which remain weakly identified by the steady-state data. The ABA hydropassive hydroactive model similarly relies on non-dimensionalization and sequential fitting to isolate key dimensionless groups governing oscillations and hysteresis. Future work should generalize these inference pipelines to include additional observables such as calcium, 2-positive and reactive oxygen species data. In this thesis, we develop a mechanistic framework that reconciles 5 decades of stomatal physiology and genetic evidence into a unified quantitative model of stomatal regulation. The fact that such a synthesis was missing for so long underscores the need for shared tools, so similar gaps do not persist in the future. One avenue is to develop open-source inference tools that allow other laboratories to fit HPHA models to their own gas exchange, hydraulic, and signaling datasets, thereby lowering the barrier to mechanistic modeling of stomata. To that end, we demonstrate how hosting interactive dashboards for model exploration and parameter fitting, for example, the publicly available stomatal conductance modeling dashboards at https://desai-sahil.github.io/stomatal-conductance-model/ can serve as powerful tools for sharing models, visualizing fits, and engaging users with varying levels of quantitative expertise. In the current landscape of advanced AI-assisted coding and analysis tools, such dashboards provide a flexible way to couple transparent mechanistic models to user-friendly interfaces, enabling rapid iteration on hypotheses and a more accessible pathway from raw experimental data to rigorously inferred parameters. The future directions outlined above aim to extend this framework towards species-specific, unified models that integrate hydraulics, ABA, Ca2+, ROS, and photosynthesis across scales from soil to canopy. Pursuing these projects will require coordinated advances in experiments, modeling, and integration with genetics, breeding, and field-scale measurements, but offer a path toward predictive platforms that can inform the design of crops and management strategies resilient to a warming and increasingly water-limited world. This concludes Mechanistic Insights into Stomatal Regulation under Water Stress.